Time is to start, so thank you very much for coming. I am Agnieszka Pluta, and I've got an honor to be principal investigator in an European-funded grant, Synchrony in Caregiver Child Interaction Across Neurodiversity. And this is my great pleasure to welcome you during this event, From Theory to Practice with SYNCC-IN Workshop. So it's a great pleasure to see so many researchers and students gathered here, both in person and online, because the whole event is also streamed, to share ideas, explore methodologies, and reflect on the ways we can better understand and support social interactions. So let's make the most of our time together. So participate actively, ask questions, and don't hesitate to sink in during coffee breaks. And this workshop is a part of a larger project which aims to bridge the gap between theoretical research and practical applications in the field of developmental science, with the specific focus on bio-behavioral synchrony. And what bio-behavioral synchrony is? It is the coordination of biological and behavioral processes between attachment partners, not only children and parents, but also classmates, romantic partners, and friends during social contacts. And why biobehavioral synchrony matters? When two individuals, a parent and a child, this is my child and my husband, become behaviorally and physiologically in sync, they are not just sharing a moment, they are building the foundation for communication, learning, emotional regulation, and social bonding. And nowadays, we think that bio-behavioral synchrony might be an underlying mechanism of effective and healthy social interactions. So synchrony helps us to understand how relationships form and why they matter, not just at the behavioral level, but through neuronal, hormonal, and automatic processes. By investigating these patterns, we move closer to identify the core components of effective caregiving, meaningful connections, and ultimately developmental resilience. I would like also to stress out that SYNCC-IN project builds on very successful years of 4EU4 plus cooperation. So a few years ago, Faculty of Psychology, Warsaw University, started a collaboration with 3 other universities in 2 mini grants. So we collaborated with Copenhagen University, Heidelberg University, and University of Milan. We together conducted research on over 2,000 parents in 4 countries. The results of those research were published recently in prestigious European Psychologist journal. We also conducted together online seminar for students from 4 countries, and we are still doing it with pleasure. We also organized a summer school in Heidelberg, and we will also repeat it this year. And then we applied successfully for SYNCC-IN twinning project. We were also joined by University of Trento, so now 5 universities are collaborating. And in this project, we intensified our endeavor to make research more interdisciplinary, more inclusive, and more impactful. And we further plan to conduct a joint research in 4 countries, so it will be a real challenge, but I am looking forward to do it. We will further organize trainings, and this is one of the trainings that we plan to do. We will also do a summer school in Heidelberg in September. We also plan to do administrative trainings, and the final endeavor of this grant, we hope that it will be a virtual lab for biobehavioral synchrony, which is aimed to be a network for researchers and teachers, educators interested in social interactions with a specific focus on bio-behavioral synchrony. So this lab will be not only to study children, but also adults. So if you are interested in following our updates, please check our social media and also our web page. And before we officially kick off this meeting, and we have very interesting plan for today, so you will learn how to apply FNIS technique, and Nirex company will teach us how to do it. We also plan to listen to a lecture by Professor Okruszek, Łukasz Okruszek, who will be talking about his very impactful research on loneliness. Then we will again learn more about FNIS technology, and then we will have a workshop. So before we officially start, I would like also to welcome our Warsaw University authorities to say a few words and welcome you. So first, we have with us Vice Rector for Research, Professor Lalak, and then also Vice Dean for Research, Professor Schuster Kowalewicz will welcome you. So the floor is yours. Yes.
Great, thank you very much. Well, welcome. Good morning, everyone. It's a great pleasure to welcome you to the University of Warsaw, which is a great center of social sciences in Poland, not only of social sciences, of course, of hard sciences as well, humanities also, but today it's my pleasure to welcome colleagues. I can see many young faces, so the pleasure is even more pronounced because I can see our future collaborators, our future partners in many endeavors, like this one. Like the very successful project SYNCC-IN, supported by European Union. You are, I understand, you are all members of the team which works in the project, and I am astonished by the number of successes you have achieved by now, and I am pretty sure that the project will be conducted to a successful completion. With many more successes, with many more scientific discoveries, which are going to be published in the most renowned journals. I'm also happy because the topic seems to be not only exciting from the scientific point of view, but very important from the societal point of view. And this is the goal of the University of Warsaw, to become useful for the society, to play more prominent, more and more important role in decision making, in creating various social and society related policies in the country and perhaps outside, to influence strongly our future, to make our lives and lives of future generations better, more successful, more impactful, and more satisfying to everyone involved. So this is great pleasure also because I understand that the plans of the team which works on the project are far-reaching. I understand that one of the strong points in the future plans is the creation of special lab in the new building, which is going to be the place of psychological research, of the research in the domain, in the area of psychology in near future. And we are hopeful, knowing that the presence and activities of that lab, partially based on the achievements you have achieved so far within the project, that the impact that laboratory, and its activities are going to be truly important and truly impactful, not only in academic sense, but also in the societal, in the domain, well, in the area of societal impact. So this is all very encouraging and very important, and University of Warsaw is going to support as strongly as possible all those activities. Well, at the end, I should mention that I am very proud that the project is based on the collaboration between various teams from 4 European universities, members of the 4EU Plus Alliance. At this point, the Alliance actually counts not 4, but 8 members. So I hope that this scope of the activities of your research will extend to our other members of the future European University based on the 4EU Plus Alliance. And well, it's great that you decided to convene in Warsaw, that you came to Warsaw, that you are becoming the part of the well, University of Warsaw community. I hope this is going to be a nice and successful meeting. Well, I also hope that you will find time to do little sightseeing around. I mean, you are in the center of Warsaw. If you are for the first time here, well, take the opportunity to visit historic places which are located here, but also nice pubs and other social centers of social activities which are located nearby. So all the best. Have a nice and successful meeting. Thank you.
Hello, dear participant. We are here because we collaborate. Our sociability, our need to aggregate, connect, form groups are the basic of essential power which make us different. The extent the capability to collaboration. We are still far from understanding various aspects of this phenomena. Researchers began examining the human mind in the real social interaction and to develop theories about mental and neuronal processing forming social brain. So brain and mind have become the idol of modernity and the subject of interest of many sciences, including psychology. This is facilitated by dynamically developing technology that makes it possible to record neuronal manifestation of human social activity. The past couple of decades produced increase of interest in interaction synchrony. Interpersonal synchrony is crucial construct in understanding social interaction and fundamental characteristic of social exchange in the synchronization of individuals' behavior, physiological responses, and neural activity. This brings us to our project. Synchrony in caregiver-child interaction project. This project, led by scientists from Department of Faculty of Psychology, brings together leading researchers from 5 institutions across Europe: Italy, Denmark, Germany, and Poland. An important goal of this project is to train participants to use sophisticated technology to study bio-behavioral synchronization and children caregivers. It's our pleasure to visit and host you to our university. I wish much you most inspiration, fruitful discussion, and effective training that will help you to discover the fantastic mystery of synchronization of human functioning. Thank you.
Thank you very much. Okay, so I think we are ready to start the first lecture given by NIREX company. So please, the floor is yours.
Here we go. Okay. Thank you very much, Agnieszka. And good morning, everyone. I'm sure I can speak for the both of us. We are very, very excited to be here. My name is Dalila Burin. I am a scientific consultant at NIREX, and I am here today with my colleague, Franziska Keller. Allow me to start by very briefly introduce us. We are NIREX Medical Technologies, a company based in Berlin, but we have a network of partners that extends basically all around the world. We specialize in providing high-end FNIRS, functional near-infrared spectroscopy solutions, to researchers such as yourself. And we do not only manufacture, of course, the hardware part, but also the software, and we do provide, of course, all of the support that you may need in your research activity. I and most of us actually in the scientific consultant team, we are former researchers, so we know the struggle, we know the pain that you have to endure, and so our job is to make your life as easy as possible by providing our solutions, our FNIRS solutions. So we give you the, let's say, hardware and software part, but you provide the most important and complicated part, the scientific part. At the end of the day, we all have the same goals, which is to make good science. As I said before, we are former researchers. I am a cognitive neuroscientist. I did my PhD back in Italy at the University of Turin. And basically in the last 6-7 years, I've been working in Japan, in Dublin, and then I moved to Berlin. And I kind of specialized in brain imaging, in particular FNIRS, and in particular in combination with virtual reality. As I said, today I'm here with my colleague, Franzi. Would you like to introduce yourself?
Absolutely. Yes, I can only add to Dalila's excitement here. We're really excited to contribute to this great workshop and to hopefully show you all how FNIRS could be a part of effectively assessing bio-behavioral synchrony between participants. I am also a scientific consultant at NIREX, have been for 2 years. I completed my studies in Germany, actually in clinical psychology. And afterwards, I worked with EEG for a very long time, but have strayed away from that. And for the last 2 years, have ventured into the exciting field of FNIRS, which is still not quite as known as EEG. So that's why we're here today, to introduce the technology to you and to show you how it can potentially be used in your research. So I'm going to give the floor to Dalila, who's going to do the introduction to FNIRS, and I'm going to be later on talking about how we can integrate FNIRS in hyperscanning studies or studies involving or examining biobehavioral synchrony. So Dalila, the floor is yours.
Brilliant. Thanks very much, Franzi. Actually, does this work? It should, right?
There's a small button I can have. Can I have it?
Thank you.
Yeah, and then it needs a few seconds.
Yes.
Yeah, it's
wonderful.
working.
Thank you very much. I have the feeling if I stand behind the podium, you're not going to see me because I'm so terribly short. It's my fault. let's start exactly. I hope everyone can hear me. I hope online everyone can hear me. If not, please attract my attention and we'll get back to the podium. Let's start from the very beginning. Let's look at the very own definition of FNIRS. What does it stand for? F, functional. It means we're looking at the functional activation rather than the structural one. Although, of course, the structural component is also very important, but it always has to be somehow accompanied by a functional activation. NIR stands for near infrared light, which is the specific type of light that FNIRS uses, and we're going to see later in the presentation which kind of light specifically we use. S stands for spectroscopy, which is the study of the interaction between light and matter. In total, FNIRS, which stands for Functional Near-Infrared Spectroscopy, is a form of non-invasive optical imaging that measures the hemodynamic changes at the level of the brain cortex. But what are specifically these hemodynamic changes? When a person is performing a certain task, whatever the task is, it can be a motor task, a cognitive task, whatever, there is an increased neural activity in the specific portions of the brain that are directly related to the execution of the task. That means that neurons need more energy, more metabolism. Of course, neurons don't have their own reserve of energy, so they have to take it in the form of oxygen from the bloodstream. So essentially, the system responds with the so-called neurovascular coupling by vastly increasing the local supply of fresh blood flow with oxygen in the portions of the brain specifically related to the execution of the task. So what happens essentially is that the oxygenated hemoglobin is a protein that sits in the red cells. The oxygenated hemoglobin that contains the oxygen along its path, once it reaches the necessary portions of the brain, releases the oxygen, and therefore, once deoxygenated, continues its path back to the lungs. This is precisely what the hemodynamic response is, this specificity in terms of temporal and spatial specificity of fresh blood flow, oxygenated and deoxygenated. And this, the graph on the left side of the slide, this is how, let's say, a perfect, clean pattern of a hemodynamic response looks like. Now, let's make it slightly interactive, because I'm already bored of listening to my own voice. Let's look at this graph. Now, even if you are not FNIRS researchers or FNIRS experts, what kind of information can we extract from this picture? Even if you think it's the most obvious thing, what do you see in this picture, in this graph? Anyone? Give it a try. Right, first of all, it's very obvious that there is this kind of like increasing pattern, like a bell, that then goes back to its original baseline. Very good, exactly. So you see that actually there is a sort of like starting level, which is a, let's say, baseline level, and then at some point there is a response, an increase in the hemodynamic activity. One very important thing is the timeline. So if you see, for example, at T0, there is the beginning, let's say, of this increased response activity, but then you see that it takes a few seconds, actually 10 seconds, about 10 seconds to actually reach its peak, and then it goes back to its original baseline. This is very, very important to consider. Are there here EEG researchers? A few, right? Very good. You probably, if you were looking, I'm sure you know, if we were looking at an EEG graph, probably we would be looking at something in milliseconds rather than seconds. But the hemodynamic response, because of its nature, it's relatively slow. It peaks around 6 to 12 seconds after the onset of the stimulus. This is something very important to consider for when we, for example, design an experiment with FNIRS. And we'll see later in the presentation how and why this is so important. Thank you very much. You just won a NIRX mug. Anything else? Well, for example, we have 3 lines. Why 3 and not 1? Sorry? To compare what? Very good, fantastic, exactly. I mentioned before that we have actually different chromophores, oxygenated hemoglobin and deoxygenated hemoglobin, which is already an important distinction, of course. FNIRS measures both. And it's important actually also to consider them according, for example, to your experimental purposes. You may want to focus on one or the other or both, and that's why there is also the total hemoglobin concentration. Thank you very much, wonderful, yes. So you see already, just looking at this graph, we already have quite a few important information regarding the hemodynamic response that we measure with FNIRS. I mentioned before that spectroscopy is the study of the interaction between light and matter. When we send a white light, like a incoherent source of light, which can be literally any kind of light, including the ones that are illuminating our room right now, when we send this light through a prism, we know that this can be split into the different wavelengths, into the different colors. Let's say, for example, that we want to select one in particular, for example, the green wavelength over there. If we send this specific wavelength into a substance and we read it out on the other side, the difference in terms of quantity and quality of the light that's being sent in and the light that's being read out on the other side can tell us important information about the structure that's in between. In the specific case of FNIRS, the light absorption actually reflects the hemodynamic response, which is precisely, as we said before, what we need in our FNIRS. But to specifically understand this process, we actually have to look at how photons, which are the light particles, how they propagate, how they travel. When, as I said before, when there is a source of light that can be literally any kind of source of light, including the light that we use in our FNIRS device, photons, they propagate. They shoot off of the light source and they spread, they go literally everywhere. Essentially, these photons, when they have to pass through a tissue, are subjected to 2 particular processes, absorption and scattering. Absorption is when a photon hits a molecule, it's destroyed, releases its energy, and therefore it doesn't exist anymore. It cannot go through the tissue anymore, of course. On the other hand, scattering is when a photon refract, reflect, or diffract off of and through the tissue. That's when our detectors come in play, because they detect, literally, the photons that are not absorbed, that are scattered on the other side of the path. Now, here we have the first problem. We know that, for example, for one photon that is detected by the detector, we have to send in about 1 million photons, which means to have a sufficient amount of photons detected on the other side of the path, we need to send in billions and billions and billions of photons. But there is another problem, the scattering process. We cannot completely account for the scattering process. As I said before, photons, they shoot, they go everywhere. So we don't really know exactly where they go, of course, but we can somehow control and predict the path that at least the photons that reach the detector, we can, for those photons, we can actually somehow predict the path that they take. And this path is actually that 3D shape, the yellow shape that you see, that it's basically probing in between and underneath the source of the detector. This shape is actually called photon banana. I know it doesn't sound like it, but it's a scientific term, and I love it, so I'm gonna repeat it many, many times today. Bear with me, please. Basically, you see the photons that reach the detector, we know that they actually travel in this kind of like particular shape, probing literally the surface of the brain. Yep. But what are the specific wavelengths, the specific type of light that we need to use in FNIRS to selectively detect the hemoglobin concentration? If we look at this picture, you see that we have biological tissues, the hand and the skull. On the other side of the picture, someone is sending in white light, an incoherent source of light. And we see that on this side, excuse me, on this side, the light that we see is red, which means that red, probably long red, wavelengths are the ones capable of passing through the tissue. But which ones exactly? Which ones specifically? To answer this question, we need to look at how the different wavelengths of light are selectively and specifically absorbed by the biological tissues. If you look at this graph, we have on the x-axis the different wavelengths. And on the y-axis, we have the so-called absorption coefficient, which is essentially how good each molecule is at absorbing light. Now, let's target more or less, you see, we said we have to look at red, probably, wavelengths, because we see that they can pass through the biological tissues. So let's target more or less the range between 700 and 900 nanometers. If we look at specifically that range, we see that there is actually a peak of activity. There you go. There is a peak here of absorption for the deoxygenated hemoglobin, which is specifically here at 760 nanometers, while we have a peak of absorption for the oxygenated hemoglobin at 850 nanometers. That's why our FNIRS, just like the vast majority of the FNIRS available in commerce, they do specifically use these two wavelengths, 760 and 850 nanometers for deoxygenated and oxygenated hemoglobin respectively. You see that, for example, if we go over 900 nanometers, oh no, I didn't mean to do that. There you go. If we go over 900 nanometers, water starts to significantly absorb light. And of course, biological tissues are mostly made out of water and lipids, so it's a problem. That's why we use these two wavelengths specifically, because we can safely and specifically target oxygenated and deoxygenated hemoglobin only. I briefly mentioned before that in spectroscopy, when we study light, we almost always have a source of light that emits light and a detector that receives it. In the case of our FNIRS, actually we have a very nice example here. In the case of FNIRS, sources and detectors are called optodes. The couple, let's say the pair composed by one source and one detector, is called a channel, which is an essential unit in the FNIRS composition. Now, the channels they detect the light that is scattered in between and underneath the distance between the source and the detector. You remember the photon banana before? This is exactly what happens in the channel, okay? Now, there are a few very important considerations to do. First, the depth of the photon banana, so how deep the photons actually go. We know that the distance between source and detector can affect significantly the path that the photons actually take. For example, if we have our source and our detector very far apart, it's the photon banana will actually be very deep. It will go deeper down in the brain cortex, in the brain regions. But of course, if you space too far apart sources and detectors, you will have a signal that's mostly dominated by noise because not enough photons will actually reach the detector. By contrast, if you place source and detector very close together, you will have surely a very high signal to noise ratio, but also a shallow depth of image. So for example, the photon banana will not go very deep into the brain cortex. Generally speaking, this very much depends, of course, the configuration you want to use in your FNIRS design very much depends, of course, on the purposes of your experiment. But generally speaking, in the adult brain, a typical configuration, a typical distance for sources and detectors is about 3 centimeters. In this way, we can actually ensure that the photon banana will go deep down into the surface by 1.5 centimeters, which is half the distance between source and detector on the surface of the scalp. In this way, we actually know that the photon banana probes just enough to go through the surface, the cortex, the surface of the brain. While usually for children is a little less, of course, because you have to consider the different head size, you have to consider the difference of the thickness, of course, of the skull. So it's usually between around 2 centimeters, something like that. But again, it can depend on the experimental purposes and the population, of course, of reference. No, I forgot to say something. This is a typical configuration, as I said, so having your sources and detectors 3 centimeters apart. In the adult brain, around 1.5, 2 centimeters in the kids, in the children's brain. But of course, this configuration, when you place your sources and detectors all at the same distance, it doesn't consider the depth of imaging. That's when tomography comes in. So when you essentially, and on purpose, put your sources and detectors at different distances from one another. So you're gonna have different depths of images, different depths of the photon banana. Now this is actually not a very common strategy. Most of the publications and the studies with FNIRS, they do use a typical topographical layout, so with sources and detectors at the same distance, because it's generally very easy to implement, very easy to process and to analyze. While if you have sources and detectors at different distances, it can make things a little complicated, so it's not very, very commonly done, with one big important exception: short channels. Now, we said before, and I was actually not completely honest with you, we said before that in the photon banana, photons travel from the source, they probe the surface of the brain, and then they get part of them, at least some of them, detected by the detectors. The truth is that in between this path, there are actually different layers. For example, the first layer that the photons have to go through is the skin, and then there is the skull, the bone, and then they actually reach the cortex. Now, we don't really care, of course, for everything that happens here, outside of the brain cortex, because this is not task-related hemodynamic activity. But there is, of course, blood flow there, obviously, because I assume your participants are alive and they have a beating heart and they're breathing, which is also very important to acknowledge. And so in the extracerebral part, there is, of course, some sort of systemic physiological activity that can mimic or affect significantly your recording. So what we do is that we place sources and detectors very close together, like here, you see. This distance actually between the source and the detector actually is 7 millimeters, which is a very, very short distance to achieve for a hardware system, and it's just enough to make sure that the photon banana actually detects only the extracerebral hemodynamic activity, not really the systemic physiological activity. So when we record from the short distance detectors, we can actually selectively process what happens outside of the brain, outside of the cortex. And therefore, when we process our data, we can regress out these measurements. So we make sure we have exclusively and only the hemodynamic activity that happens here, which is exactly what we want to record. Just to give you an idea of how important these measurements are, this graph here, you see, I hope you recognize practically kind of the same hemodynamic activity that we saw in the very first slide, right? It looks almost exactly like that. It's just a little dirty, it's not like a perfect, clean curve. This is because this measurement still includes the short distance detector, short distance channels. So they are still contaminated by the extracerebral hemodynamic systemic physiological activity. This is the very same measurement with the short distance channels regressed out. And you see that the measurement is much more similar to the perfect pattern, let's say, that we saw at the beginning of our presentation. So this is important to understand. What we measure when we record the hemodynamic activity from source to detector is kind of like everything that happens in between, but we want to specifically and selectively measure what happens in the cortex. So we need to kind of exclude everything that happens outside of the extracerebral part of the measurement. So far, I've been practically speaking almost exclusively about light. But how do we get from light intensity to hemoglobin concentration? First of all, something important to remember is that our FNIRS, just like actually the vast majority of the FNIR systems available in commerce, is called a continuous wave FNIRS, which means that it works by emitting two constant wavelengths. Does anyone remember which ones? The wavelengths? 860. Very good. You also won a sticker and a mug. Very good. Exactly. So it works by emitting these two specific wavelengths constantly, 760 and 850 nanometers. In this kind of systems, the problem, as I said before, one of the problems is that we cannot completely account for the scattering process because we don't know simply the path that all of the photons take. To bypass this problem, FNIRS, continuous wave FNIRS, do not measure absolute values, but rather concentration changes in the in hemoglobin concentration. Generally, it's the reference the measurement uses is either a period out of the real measurement, let's say, or sometimes an average of the entire recording. But anyway, the important thing to understand here is that FNIRS, continuous wave FNIRS, does not measure absolute values, but relative changes in hemoglobin concentration. So to go from light intensity to hemoglobin concentration, the first step is to determine the optical density, which is essentially the negative logarithm of the raw light intensity over the mean light intensity, raw measurement, of course. So essentially, it's a kind of like a standard, a normalized measurement of the light intensity. For the record, this formula, and also the second one I'm going to show you, for educational purposes today, we're going to go into details and understand what's the process behind those. But in the vast majority of the softwares for data processing and analysis, it's one click. So you don't have to worry about, for example, remembering the formula, but it's important to understand what happens. So as I said, the first step is to determine the optical density. And the second is the modified Beer-Lambert law. Again, sorry. Again, this formula looks a bit scary, but trust me, it's not. And again, it's literally just one click. We are going to explain how it works just for, of course, educational purposes, but you don't have to remember or learn it, just to understand essentially the process behind that click, let's say. So this Beer-Lambert law, or better, the modified Beer-Lambert law, because as I said before, we are not measuring absolute values, but concentration changes, works essentially in this way. We first have already our optical density here. Yep. And it's delta just because it's, as I said, the relative changes, not the absolute values. Lambda is the specific wavelength, which means that basically this formula has to be repeated twice for the two wavelengths. The epsilon here is the so-called extinction coefficient, which is essentially value of how good each molecule is at absorbing light at a specific wavelength, lambda. Don't worry, it's a constant value. I don't even remember, to be honest. Like, for oxygenated hemoglobin is 1.79 milliliters, inverse milliliter per molar. And for deoxygenated is 120, I don't remember, something like that. It doesn't matter because it's a constant value. We're going to ask ChatGPT later. But just to remember that this is the first important factor in the formula, the extinction coefficient for the specific chromophores, oxygenated, deoxygenated hemoglobin. D stands for the distance between source and detector at the level of the scalp. In the typical configuration in the adult brain, that is very good. Another mug over there. Remind me, please come over here later and I will give you all of the mugs that you want. Exactly, so 3 centimeters. In the children's brain, very good, exactly, 1.5 to certainly less than that. Brilliant, exactly. Last but not least, the differential path length factor here is simply a multiplication factor that accounts for the fact that the photons don't go straight from source to detector, but they take the famous photon banana, which means they travel much more than the actual distance from source and detector. Again, it's a constant value. It's typically, if you have a source detector distance of 3 centimeters, it's typically 6 or 7, which means that photons travel 6 or 7 times more than the actual distance between source and detector. So far, everything okay? All good? Wonderful. Am I on time? Yes, fantastic. FNIRS, of course, is just one of the many brain imaging modalities. I heard before that there are a few EEG researchers here. I know that there are fMRI researchers, right? Right? Fantastic. Yes, brilliant, exactly. So how does FNIRS compare with the other brain imaging modalities? In terms of spatial resolution, the gold standard, let's say, is generally considered to be fMRI, with, of course, a resolution of about 3 millimeters, which, of course, in terms of spatial resolution is extremely high. And, of course, fMRI can basically scan cortical, but also inner structure, so basically the entire head, of course. While with FNIRS, we are, first of all, limited to the cortex because the photons cannot go too deep into the brain. They just probe the surface of the cortex. And generally considered the spatial resolution of FNIRS is about 2 centimeters, 3 centimeters, although you can actually, as I said before, space sources and detectors closer or slightly farther apart to actually achieve a high density, what we call then high density montage, which means that we can actually increase the spatial resolution of our FNIRS, bringing it to almost an fMRI level, which is actually very exciting, and it's becoming really, really popular in the last years. FNIRS, you can see we have an example, a few examples over here, kind of looks like an EEG, more or less. And actually the setup is relatively similar to the EEG, to an EEG configuration, although FNIRS is generally much more robust to motion. That's why it's very commonly employed in very ecological setup and in situations where there is a lot of movement, lots of action. Also, it doesn't require any gel, any water, because it's light, so we don't need any additional water, which is generally very appreciated by participants. And in terms of temporal resolution, actually, as I said before, something important to consider is the timeline of the hemodynamic response. The EEG has generally a very high temporal resolution, while FNIRS, we have seen before, because of the nature of the hemodynamic response, is relatively slow. It depends, of course, what kind of tasks you're looking at, but it's generally slower. Although it's actually a little bit better than an fMRI, which doesn't have generally a very high temporal resolution. So as I was saying before, there are several advantages using FNIRS. First of all is very easy to set up, very quick. For those who are going to join our hands-on experience in the afternoon, you will see actually how quick, easy, and fast the setup of the device is, which makes it very suitable for certain populations where you cannot afford to spend too much time with them, like children or neurological or clinical populations. It's also very suitable, as I said before, with situations where there is a lot of movement, or, for example, with kids where you cannot really force them to stand still, although I would really like to do that, but it's not ethically allowed, I think. And it's extremely, obviously, portable, just like an EEG, which makes it very suitable for a very ecological situation. You can complete your data collection in a very ecological setup, or a social context, a social situation. For example, hyperscanning, meaning you can record from more than one participant at the same time while they perform social tasks or whatever, which is actually what we're going to see today in the hands-on experience. What can we do with FNIRS? The sky is the limit. It's, as I said before, very suitable for certain populations, of course, young adults, but also elderly, clinical populations, neurological populations, or children, of course, because as I said before, the setup is very convenient, quick, and easy. As I said before, it's also very suitable for ecological and social interaction situations, such as, for example, hyperscanning. Or it's very commonly used, for example, for BCI, brain-computer interface, language process. I heard before that someone, I remember, is doing like language research, auditory or cognitive, in general, cognitive processes, motor components, of course. And it's also, interestingly, very suitable for multimodal recording. For example, FNIRS and EEG at the same time, or even FNIRS and fMRI, or TMS, or physiological measures. We also have our physiological measurement device, which we're also going to see today in the afternoon for example, heart rate, skin conductance, temperature, and so forth. It's also perfectly compatible with other modalities, such as eye tracking or virtual reality. As I said before, this is actually one of my expertise, and this is one of the things I've worked in the last years back in my research days. So to very briefly summarize, yes, we have seen how the hemodynamic response works and looks like, how we go from the light, the near infrared light that we send in the cortex, and how we read it out on the other side, and how do we go from light intensity to hemoglobin concentration. So far, everything good? Questions? Problems? Please. Yes, very good question. The question is if the continuous wave FNIRS emits two wavelengths at the same time. Yes, very good point. Specifically, yes, we have these LED systems. We have different sources of light, but generally our system and most of the systems available, they have LEDs sources. And they do emit constantly these two specific wavelengths at the same time. Although they do not emit light all at the same time, because the problem is that there might be a problem of crosstalk between channels. So for example, if you emit light from one source, the problem is that, as I said before, light scatters. So the detectors may detect it from here, 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 here. So you don't know if the measurement that you get is actually what you're recording from this specific channel. So we use this particular system called multiplexing, which is essentially a way of turning on the sources at different time points. So for example, this source start first, let's say, and also this source here in the back, because they are far enough, so there is no chances essentially that the detectors may get confused and may record, you know, from those two sources. This is a problem, for example, when you have a high density montage or a very high coverage, like a full head coverage, because there are lots of chances of crosstalk. So we avoid it by turning the sources, not all of them at the same time, but in a kind of like smart way to avoid essentially the crosstalk. So they do emit the wavelengths at the same time, but the LEDs are not always active at the same time, but not because of the wavelength problem, because of the eventually the crosstalk problem. Please. I'm not sure. I really hope one day we will find a technology that can actually detect just optically inner structures, not only the cortical structures. As I said before, at the moment, this is a limitation, of course, that we have that we need to consider. But generally speaking, most of the researchers, of course, using FNIRS, they do target tasks, especially cognitive or motor tasks, that are generally very conveniently and easily recorded from the surface from the cortex, obviously. I don't really know. I sincerely hope that one day we will get there. But for the moment, this is what we have, yes. All good? If you have more questions, you're very welcome to ask later, to contact us directly. But please go ahead. True. Yeah. I can tell you actually more than that. We're already doing it. We have a lot actually of researchers. We know several researchers, and we know that there are a lot in the world. Researchers about like some movement science or sports science, that they do record in very ecological situations, like for example while running or while performing physical activity, and perhaps even like in like a real multiplayer situation. I cannot think of a publication right now, but if you give me some time, I'm gonna find something and maybe we can send it to you. But I know for a fact that it already exists, totally. That's actually what makes this technology very suitable for this kind of research. The fact that it's very robust to motion, even in a very highly moving situation, and that can record in very ecological context and from different subjects at the same time. Absolutely. Sorry again? Wirelessly. Oh yeah, absolutely, yes. We actually, for example, will see. Are you coming to the hyperscanning hands-on in the afternoon? Oh, okay, then. But yeah, anyway, yes, absolutely, yes. Actually, we have essentially two ways of connecting our devices. Okay. Very kind, Agnieszka. Very kind, Agnieszka. Absolutely, yes. That's actually what makes possible recording in very highly situations with high levels of mobility, of course, yeah. In our case in particular, we have essentially two ways of connecting the device and recording data. With USB cable or wireless. If you have a static situation, like a typical, I don't know, cognitive task or like a monitor, you know, desk or whatever, you can go for the wired connection and you're totally good to go. But if you need movement, or perhaps your participants need to be more comfortable and you want them to make sure, you want to make sure that they are actually interacting in a normal social situation, you can totally go for the Wi-Fi. That's exactly what's designed for, yes. Thank you very much. Okay, let's continue. I should be relatively on time, wonderful. I'm going to share we are going to share all of these slides with the participants later, right, Agnieszka? So you don't need to, you know, write down everything here. There are a few useful publications and resources you can have a look at if you want to explore some of the concepts I briefly described more in detail. Let's move on now to the second part of the first part of my presentation. Am I correct? Like, am I right? Perfect, wonderful. Let's look now at how to design an experiment with FNIRS. Now, there are a few important considerations to remember when we design an experiment with FNIRS. Most of them, actually, I briefly mentioned them before in the first part of my presentation, but it's very important to consider why they are relevant for the experimental design. First, as I said before, it's very important to consider that with continuous wave FNIRS, we don't measure absolute values, but concentration, relative changes in the hemoglobin concentration. Another very important thing to remember is that we cannot completely account for the scattering process. So we don't know, for example, the time of flight of the photons, which means that essentially what we have is a sort of like depth estimation, essentially, of the photon banana, hence the source and detector separation. And of course, as we said before, and it was also highlighted in one of the questions, we have to limit, excuse me, we have to limit our functional tasks, functional activations to the cortex. We, at the moment, cannot go deeper into the brain, so we need to target brain regions that are activated by tasks that can be visible essentially from the cortical level. Also important to remember, as I said before, the spatial resolution. We generally speaking are looking at 1-2 centimeters of spatial resolution, much higher if you have a high density montage. So when you space probes, sources and detectors either closer or slightly farther apart, you can actually achieve a much higher spatial resolution. Almost to an MRI level, actually. Also important to consider the inter-subject variability. It's sometimes a bit high, actually, partially because the hemodynamic signal, the FNIR signal, is actually quite different between one participant and the other. Although, of course, there are several strategies, especially in the processing phase, that you can actually have to make it as standardized as possible. Another considerations is that, especially for your setup, for your experimental setup, is that the detectors, especially in the FNIRs, they always detect light, which means that they may easily detect light that doesn't necessarily come from the surface, but from the environment. This is also, of course, very important to consider when you design your experimental setup. You want to make it ecological, you want to make it a naturalistic environment, but there are, of course, lots of sources of light in the environment. We can somehow easily actually control this problem by, for example, wearing an overcap or something that can mask the environmental light. But still, it's something important to consider. Another thing I said before, that FNIRS is generally very robust to motion artifacts, but of course there are situations where, especially when there is a lot of natural movements, like imagine like a sports situation, motion artifacts are inevitable. You're gonna end up sometimes with a little artifacts in your recordings. Again, there are solutions to this problem, in particular in the pre-processing part of your analysis, but again, it's important to remember and to consider. We already mentioned before the extracerebral physiological activation, which is inevitably part of the hemodynamic recorded signal, but we already said how we can somehow regress it out and having a clean, pure signal of the hemodynamic response coming directly from the brain, the task-related hemodynamic activity. And finally, last but not least, the setup time. Of course, the more coverage you have in your montage, so the higher the number of the optodes, the higher probably is the setup time. But even, for example, in the case of the cap you see over there, which is essentially a full head coverage, it definitely requires not too much time to set up on the participant, much less, for example, than an EEG, which makes it, as I said before, very suitable for certain populations, like children, for example, where you don't want them to waste too much time, let's say, waiting for you to prepare the cap. That said, of course, as always, these are kind of like general, let's say, considerations when designing an experiment, and you are the expert of your research, so I'm definitely not gonna tell you what you're supposed to do with your research. It's just important to remember and consider that there are a few important things to remember when designing an experiment with FNIRS. But just like any other research, and definitely you are, of course, the expert in this sense, everything starts with a clearly defined research question, obviously, which can start from your knowledge, someone else's knowledge, publications, your ideas, whatever you can come up with, your curiosity, whatever you can come up with. And therefore, consequently, you may want to target, for example, a specific population, you may want to use a specific task, or you want to target a specific portion of the brain. Sometimes, for example, quite commonly, researchers have already an idea in mind of, for example, what region of the cortex should be, in theory, activated during the execution of a specific task, and therefore they already target that part of the brain with the FNIRS. Or it can be more you can use a more explorative approach and, for example, use a full head coverage and explore different regions, different targets. You may not be super familiar, or maybe you are actually, with some of the data processing strategies, for example, general linear models. But it's also very important, even in the phase of experimental design, to already consider which kind of data processing, which kind of analysis are you going probably generally to apply to your data once collected. Something very important I would like to say here is actually the frequency of your task. I said before that the timeline of the hemodynamic response and therefore of the FNIRS recording is very important to consider because it's relatively slow and we need somehow to adapt our experimental design, our task, to this constraint, let's say. What you don't want to do is, of course, to filter out when you process your data, the frequency of your experimental design, your experimental task. So what you want to consider, excuse me, we're gonna see immediately a couple of examples of it, but what you want to consider is, for example, to have a period where you have the hemodynamic activation, so let's say the active part of your task, followed by another part of rest. As we said before in the hemodynamic response, this allows to the activity to peak and then to go back to its original baseline. This is important to know and to calculate because then when you process your data, you don't want to filter out precisely the frequency of your task, of course. I said before, of course, many times that the most important, the crucial measurement in FNIRS is the hemodynamic response. Now, the brain, or better, the blood flow, because the brain does not respond instantly to the stimulus, or better, it does, but in terms of hemodynamic response, it takes a few seconds to actually peak and then to go back to the baseline. So, when we analyze the data, when we process the data, what we do is actually to model the time course of the hemodynamic response. And we use a mathematical process called convolution. We literally convolve or combine, let's say, this kind of canonical typical response shape with the frequency of your experimental design, with your stimulus. Think of it actually literally as a formula. So you have your stimulus, whichever it can be, times the response shape of the hemodynamic response, and therefore you get an expected signal. This is actually not that different from what happens in an fMRI processing. Now, how to design actually an experiment with FNIRS. This depends very much, of course, on the purposes of your experiment. So, again, you are the expert of your own research, and you have to consider, of course, what are the targets, the purposes, and the goals that you want to achieve. But generally speaking, let's say, there are, two main strategies to design an experiment with FNIRS. The first being the block design, good old block design, where essentially you have blocks of trials where you repeat, let's say, or maximize there the chances of having an hemodynamic response. So a block where the participants generally repeat the same task, and there is where you actually want to see your peak, your increased hemodynamic activity. This block is generally followed by a resting period, so we allow the hemodynamic response to go down, and we basically match the typical, let's say, hemodynamic time course, timeline. Generally, the blocks, they go from 2 seconds to 50 seconds. It's something we see in most of the publications in literature, and we also recommend, is that the resting period should be at least the same amount of time of the block period. So for example, if you have a block of 12 seconds, you should have a resting period of also at least 12 seconds. Something else we actually we know, because some smart people in literature, they already told us, is that we generally recommend to avoid blocks of exactly 10 seconds, because 10 seconds exactly match the Meyer's waves, which are very slow, relatively slow physiological frequencies generally associated with breathing. And they go exactly at 1 hertz, right? Which means that they actually match, they can mimic the hemodynamic response of the block. So we generally recommend to avoid blocks of precisely 10 seconds. Go for example, 12 or 15 or something like that. The block design is probably the most common experimental design for studies with FNIRS, because you can really maximize the chances of having and seeing a very strong hemodynamic response. It works very well in general with the FNIRS measurements. But of course, it's not always possible. There are tasks that cannot be performed in a block design, especially when you have a problem of, for example, repetition effects or learning or predictability, these kind of things. So in this case, we can actually do an event-related design. This is probably design strategy that is much more common to EEG researchers. It's also, of course, very commonly used in psychology or cognitive psychology, especially for certain tasks, I have to say. In this case, of course, you have very short, much shorter trials, for sure, respect to the block design, where actually you don't even have a block, you have the single trial, for example, repeated kind of like independently from one another. And of course, a certain resting time, a little separation in between one trial and the other. Now, in this case, because of the relatively slow nature of the hemodynamic response, it's possible that the hemodynamic, actually it's very likely that the hemodynamic response will overlap between one trial and the other. But with a few simple strategies, we can actually control and avoid this problem. The first being, if we simply introduce an, let's say, randomized or different interstimulus trial interval, we can actually very simply control a little bit for this problem already. So for example, instead of having trials separated by always the same resting period, we very strongly recommend, and the studies in literature confirm, to jitter, to introduce a randomized, for example, interstimulus interval, which is generally relatively simple to achieve. Something else, of course, we can do to control for the overlapping of the hemodynamic response is in the data processing phase. Generally, there are several strategies, including especially general linear models, like I mentioned before, where we can actually associate specifically the activity peaked over a specific trial or another. So in this case, even though the hemodynamic response is relatively slow, and it is, we can actually have an event-related design study with FNIRS as well. There are rules, let's say limitations, something we have seen from different studies in literature. It seems like there should not be less than 2 seconds interval between one trial and the other. This is the only limit, the only constraint we generally recommend when applying an event-related designs for your FNIRS study. There is a third option, which is a sort of mix between the two strategies, so block design and event-related design, where you have, for example, blocks of events, but inside the block you have, for example, 75% of one trial, let's say, and 25% of the other. A very simple case is the go-no-go task, just to give you an example, and probably a task that everybody more or less knows. In this case, you actually have generally a very high signal-to-noise ratio because you have the block design, which maximizes the possibilities, the chances of a strong hemodynamic response, but you avoid the problems of, for example, predictability that you generally have in a block design experiment. Very briefly, just to say that there are other experimental design practice that you may want to consider according to the research you want to do. They're becoming, as I said before, very popular, these, let's call them real-world design, where, for example, you want to apply your recordings in a very ecological and very naturalistic situation. For example, in a hyperscanning situation, when you have more than one participant interacting and performing the task or whatever at the same time. It's also becoming very popular, the resting state, all the default mode network recording. So essentially to record when the participant is not performing anything in any specific task, let's say. So it's a sort of like resting activity measurement, which is becoming actually more and more popular, I have to say. Last but not least, functional connectivity analysis. You can actually record, as I said before, if you have enough coverage, for example, in the case of the cup over there, you can record from different brain regions, and you may want to see, for example, the functional connectivity, the functional activation of these different brain regions during the execution of a task, or between participants. So you can actually do it within the same subject or between subjects. Questions? All good? I am perfectly on time. I'm very happy about it, really, really. Okay, with that, I thank you all for your kind attention, and I think I will give the control of the stage back to Agnieszka. Thank you. Thanks very much. I forgot already where was the button. Sorry.
Yeah, I will take care of it. Is it still working? Okay, so thank you very much. It was a great pleasure.
Thanks.
And you know why now. NIREX is a leader in this field. So thank you very much for this excellent talk. And we would like also to thank you with just a small gift. Don't run away. I know that we are still have you with us, but it's just a small incentive for staying with us longer. So thank you. Okay, so now thank you very much. And now it's time for a break. So we have 30 minutes according to the schedule for coffee and some snacks. And there are some materials from our university and NIREX team. So please take them, and also badges. So you can take badges and just put your name. Like badges, like this one, and they are over there. So yeah, and enjoy the, sorry? And certificates, yes. If you need a certificate, you can write down your name and put it into this box. Your personal data will be protected. We will close the box. Then Jana will send you the certificates. Email, yes. So we will send the certificates, and especially students, you might be interested in having them. So yeah, enjoy your break, and yeah, connect with your colleagues. we continue. So hopefully you are all relaxed after the break. So let's continue. And we have with us Professor Łukasz Okruszek. You can see that there is a small change in a program. Our presenter, Dr. Czeszumski, got unfortunately ill. So we wish him speedy recovery. But we've got a great replacement, so thank you very much, Łukasz, for accepting my invitation at this very short notice. And Łukasz is a key researcher in a field of social neuroscience, and I am always intimidated by the amount of work he's doing. So he's head of a lab, social neuroscience lab at Polish Academy of Sciences. He runs successfully many projects applying different methodologies, including EEG, fMRI, heart rate variability, behavioral examination. Anything else? Did I miss anything? That's okay. To study underlying mechanism of social cognition, and his recent interest is loneliness, so a very important topic for our well-being and health. So Łukasz, again, thank you very much. The floor is yours.
Thank you, Agnieszka, for the invitation. Now I'm blushing after this introduction. So. Okay, thank you. So I'm gonna tell you a bit more about our recent 7 years old interest in loneliness research, and given the fact that it was quite an immediate, I wouldn't say sudden development for me, because I agreed to be the replacement yesterday afternoon, so beware that the two-person setup will be just in the very end of the lecture, and I'm going to start by introducing you to the whole topic and how we actually, what was the trajectory that led us to develop a bit more two-person setup to dig into loneliness research. But first, I'm going to tell you a bit more about loneliness itself, because this is like a very common word that brings many different images to our mind, and probably you have noticed, even before COVID times, that there's this little thing called epidemics of loneliness, or at least media say that there's epidemics of loneliness, and even before COVID times, all of these headlines are from 2018, so pretty dated at this point. We were able to see that there are some problematic things in terms of public health which relate to the social isolation and loneliness. And what is loneliness? Loneliness, by our psychological definition, is any situation where you feel a significant mismatch or discrepancy between the person's actual social interest and his or her needed or desired amount of social interaction with others. So there's no objective quantifier for loneliness. This is a subjective mismatch. If you feel lonely, if you're not happy with the social relationships you have, you are lonely by this definition. And this is something that is not very, very widespread knowledge, because many people connotate loneliness with elderly people who live alone, who are actually socially isolated because they have very little contact with others. And loneliness, it may be something that brings more to your mind images like being lonely in crowd, having people around me and yet feeling that it does not give you what you need. And this is not something that we developed. This is like an actual thing in social sciences. If you look at the European Social Survey, they also ask different questions about social isolation, which may be objectively quantified by the amount of social contact. How much social contact did you have in last week, 2 weeks, etc., and loneliness. So being happy or not being happy and satisfied with your social relationships. And the other thing, which is a bit problematic in Polish, which basically used the word samotny for many different states and ideas, is that we don't have this nice division. And in Polish, if you say samotny, people may have images like this. Every single person who graduated from the high school and had like the courses about romantic literature have the ideas like, but why would you study it? Like a negative thing. how much would I give to just walk in the wood on my own? It's a perfect feeling for me. How much would I give just not to sit all day long with my family? Why would you say it's a negative thing to do? And in English, there's this nice differentiating line between solitude, so basically being reasonably comfortable with one's own, and loneliness, so being uncomfortably alone without someone. So if you just like reading a book on your own, staying alone in home for the whole evening, that's okay. It's a positive thing which will probably improve your well-being, not decrease it. And this is the different thing that we study. And if we ask people, do you need other people? Do you have social support you need? Are you happy with your relationships? And they say, no, I have no good relationships. People are there, but they are not for me. It's not solitude, what we study. So, why is it the problem? And why do we say that there's a loneliness epidemic? Basically, there were several large population-based samples studies which have shown around 2018 that there are many lonely people in population of United States and in UK. So there were like these two very big studies, one by the insurance company called Cigna with 20,000 Americans, which showed that basically half of the Americans say that they are often or always lonely. There was also a BBC loneliness experiment with 55,000 UK respondents, which also have shown that around 1 out of the 3 people in Britain feel lonely and marks it as a problem. And additional thing, if you look at the age distribution, it's not elderly who say that they are most lonely. The most lonely group in both of these studies were the youngest groups, young adolescents and young adults, Gen Z, so-called. And by 2022, we also are there. There was like this large report by Instytut Pokolenia in Poland. They did the population-based study and they shown that there's actually we were able to perfectly replicate the patterns of loneliness distribution, the levels of loneliness. There's actually very similar median in our sample as in the Cigna study, and there's also this steep decrease in loneliness by age groups. But okay, maybe there's no bearing on public health and that, but probably you may have heard that being lonely is like smoking 15 cigarettes a day. Very peculiar number, which comes from the large-scale meta-analysis by Julian Holt-Lunstead and her collaborators, who did sum up 70 prospective studies with 3 million participants, and they have shown that basically both loneliness and objective social isolation have similar effects on longitudinal outcomes in prospective studies like mortality, like morbidity, as well-known health risk factors like smoking or being obese. So this is why some people may say that like if you're lonely, it's like smoking a pack of cigarettes a day. It's a very wide metaphor for that, but I would say there's something in it. And how do we explain these effects? Obviously, there are many factors which may be linked to it in the contemporary world. There's this idea that loneliness may be like, let's say, obesity. So it may be treated like a disease of affluence, so something which is linked to the way we live in the modern world, and that we are not used to in terms of biological adaptations. So what would be the factors that may contribute to it? Ask politologists, ask social scientists, ask sociologists, psychologists of different orientation. Probably they will point toward many different things. Most of the people will just say social media and smartphones. But there are people who say that, yeah, it may be linked to the modern cities, it may be linked to how we spend our time, it may be linked to the way how we live, but it may also be increasingly involved, maybe increasingly linked with social media. So there are many different factors. But myself, being a psychologist who wants to study biological underpinnings of these effects, I'm not very well qualified to tell you whether the modern cities are bad for our health. I was more inclined to investigate why loneliness, think lonely, think inadequate in our social relations may have all of these biological outcomes. So we started to look in the theory, and by the time we ended our literature review, there was one very peculiar theory about how loneliness impacts one very obvious theory created by late John Cacioppo and his wife, which is called evolutionary theory of loneliness, which is basically the most prominent field-wise theory. And they say that loneliness may be treated like hunger or thirst. So this is like a biological drive that evolved to inform us that there's something important missing. So if you're hungry, you probably should focus on getting something to eat. What should you do if you're lonely? You feel not too good about it, so you should probably go meet some people, increase your likelihood of improving your relationships. So loneliness is being this negative drive that we want to suppress and to relate with others, to be reintegrated with our group. And the part about evolutionary suggests that it may be something about our survival. Why is it important? Because in bit older times, if you were not a part of the group, you were probably very likely to be a prey of some predators, to become maybe a victim of the other group, many different things, to be exposed to the very bad conditions and external conditions. So what loneliness is also supposed, according to the ATL, so evolutionary theory of loneliness, do is to improve our cognitive mechanisms to reenact our relationships with others. So on the one hand, they theorize and suggest that loneliness makes us more vulnerable to social signals, not to miss any reconnection opportunities. But given the fact that we are also super considerate about our own survival, what types of signals would be preferably processed? Probably negative social signals. So the large chunk of the theory proposed by Cacioppo is about so-called social threat hypervigilance. So these accidental mechanisms which are linked to the fact that we are very focused on our own survival, and this way, instead of focusing on reconnection cues, we start to look for the threat cues. It may have been adaptive back in the day, but if you sit all day long, 8 hours in the office, and wonder whether people gossip about you, whether people look at you in a negative fashion, it may be quite a stressor. So they suggest that the social threat hypervigilance is the main mechanism that originate the trajectories which long-term affect your immune system, your sympathetic nervous system, increase your stress response, and produce all of the health sequelae out of it. And if you look into papers where the ETL is discussed, you may easily find pictures like that. This is like colorful blobs on the brain. They are not meaningful. They are random, just to, improve the niceness of the picture. And they say, yeah, we have so much support for the theory. Some of these mechanisms, like increased sympathetic tonus or in some abnormal inflammatory response, are well applicated. But they also include stuff like cognitive mechanisms associated with hypervigilance for social threats, or stuff about brain activity patterns which should be observed in lonely individuals. Among this support for their theory. And if you look to the popular literature, you may easily find passages like that. There's this book by Norina Hertz, which is very popular in people who start to read about loneliness. Usually they start like there. And there's like passage which comes like that. Multiple studies have now shown that in the brains of lonely people, the level of activation of the temporoparietal junction, the part of the brain most closely linked with empathy, activity decreases when confronted with the suffering of others, whereas in the non-lonely, it rises. At the same time, the lonely person's visual cortex, the part of the brain that typically processes alertness, attention, and vision, is stimulated. Just as the lonely body ramps its stress response, the lonely mind, anxious and hyper-alert, operates in terms of self-preservation, scanning surroundings for threats, rather than trying to see things from the affected person's point of view. Have you ever taken a walk in the woods and jumped back because you saw a stick on the ground and thought it was a snake? asks Dr. Stephanie Cacioppo. The lonely mind sees snakes all the time. So if you read something like that, and you're naive, you're not a researcher in the field, you feel there's like very compelling evidence for this mechanism. I would assume so. I felt like that back in the day when I started doing loneliness research. And if you look closely, these multiple studies are basically one study by John Cacioppo from 2009, where he median splitted 23 students with regard to the loneliness scores. Can't say it's the most robust pattern. There's not even a cutoff score, so I basically tried to calculate the cutoff from the graph, and what I found is that this is basically a cutoff between our first and second quartile in our pre-stratification sample. So there are like actually 2 lonely people in the group, this far right around there. What about the cognitive mechanisms? According to the ETL, angry facial displays, which are basically like very robust threat signal, social threat signal, should be preferentially processed in lonely participants. And what do studies say? If you look into the literature review about the cognitive mechanism of loneliness, you may easily find that there's like one study which actually found higher accuracy for anger recognition in lonely individuals. One which did not find any association between emotion processing and loneliness, and one which found decreased emotion recognition in lonely individuals. So we started to think, can we do better than that? And by 2019, I got like the first project, funding for the first project, when we decided to look into the mechanisms of loneliness, behavioral mechanisms, physiological mechanisms, and neural mechanisms, and to actually look into them in well-sampled, well-powered samples with most robust tools that were available to us. And we started by examination whether the social threat hypervigilance is there. My background, similarly to Agnieszka, is in neuropsychology. So I decided to look at the tasks which are supposed to measure social cognitive capacity. So basically, if you discuss social cognition in terms of neuropsychological scores, you usually look at specific processes like social perception, which in this case mean differentiating between social and non-social agents, emotion processing and emotion perception, so how well are you able to distinguish different emotions in social agents, and theory of mind, meaning intention, attribution, mental state attribution to others. And there are tasks which can be used to measure how apt are you in recognizing emotion from facial displays, from vocal prosody, attributing intentions based on the story description, based on this. This is actually a very poor test, ICE test. I don't recommend anyone to use it. Still though, not everything is available in Polish, so we also did use it for part of our research, etc., etc. And very importantly, stemming from the field of clinical neuroscience, there's also another construct which may be linked to the social cognitive capacity, but it's not fully overlapping with it. It's called social cognitive bias. So you may have very nice social cognitive capacity. You are well able to recognize angry displays, you are able to attribute mental states to others, but sometimes the story is just ambiguous. So for example, you are new at the job, you've been at the job, a new job for 3 weeks. One day, you see one of your new coworkers on the street. You start to walk up to this person and say hello, but then he passes by you without saying hello. Why did the person do it? Any ideas? Very nice and healthy attribution. So some people will say, yeah, the person was mind absent, maybe she was talking over the phone or Bluetooth Bluetooth speaker, maybe the person was taken up by something, maybe just did not see me. Okay, many different explanations. But some people will say, the person is clearly angry at me. This person has something against me. Maybe this person wants my job. Anything else like that. Many different sides to the story I already heard. And you may study stuff like social cognitive bias. You may ask, How do people attribute intentions in this type of neutral or ambiguous vignettes? You may ask them about generalized statement like people cannot be trusted, things went wrong in my life because of other people. So you may measure this generalized attribute toward negative attributions, hostile attributions to others. And guess what? If we discuss social cognitive capacity, we measured it in like 500 participants back to this day, and we found no correlation between loneliness and any of these outcomes. What we found consistently in our research is that if you discuss social cognitive bias, not objective social cognitive capacity, not angry recognition, not the way you process angry cues, perceptual processes, anything like that, we find a very nice correlation between loneliness and hostile attribution bias, social cognitive bias. But one may say that, okay, but these tasks are not there to, differentiate the subtle stuff associated with individual differences with non-clinical population. These tasks were devised to differentiate between like people with frontal lobe lesions or schizophrenia, patients with schizophrenia and non-clinical populations. So maybe you need something a bit more sensitive. Okay, so we use this nice task called facing the crowd. There's this effect described widely in cognitive psychology that you're able to detect angry faces among both neutral and happy distractors or emotional distractors. You're more accurate, you're faster in detecting them compared to happy targets among all sorts of crowds. And we were able to nicely replicate all of these results. People are faster, more accurate, there's this nice interaction, angry faster, so-called threat superiority effect. We have the data from 350 people, and guess what? No correlation with loneliness. So maybe there's something about appraisals, this perceptual appraisals. There's this thing called morphs, which may be modeled to elicit specific first impressions. Which of these guys is most threatening to you? The one on the left or the one on the right? Anyone says the one on the right? Maybe like that. Let's start like that. Anyone? So you're very congruent with most of our participants. And this is the correlation between the appraisals of threat from these guys and loneliness. You see the pattern here? I do. You may also dig deeper. You may say, okay, maybe something's happening at the neural level. So we have data from loneliness questionnaires and brain activities from like amygdala localizers, which are linked to social threat sensitivity, to all sorts of dynamic and static pictures, responses. We find nice and replicable patterns of responses and little to no correlation with loneliness. I can do it all day. We've got data from ERPs, we've got data about heart rate variability. Believe me, not much there. So can we safely say that there's not much there in terms of social threat hypervigilance? We also ask our participants to do like ecological sampling, experience sampling in our research. So we ask our participants, group of over 170 participants in this actual part of the research, to do the ESM protocol for 7 days. So they drag the phone with special app, and several times, up to 9 times a day, they were asked about, how do you feel right now? Are you lonely, abandoned, tense, irritated, etc.? And then they were asked about contextual factors. Did you have an interaction with other person? Do you have company right now? And if they say, yeah, I'm with someone right now, we ask the specific questions about their social appraisals, which were targeted. Is this company accepting, comfortable, pleasant? Is it judging, threatening, distressing? And now, spoiler alert, people do not stay at many different occasions with people who they deem extremely threatening. Not much of that type of appraisals in our sample. But if you look at the negative and positive appraisals, more lonely participants are more likely to look at their social peers in a more negative, more threatening, more distressing fashion and less positive way. So maybe all of these nice laboratory tasks do not capture something which is happening in real life. You know, basically, if we look at the laboratory experiments and we compare them to the natural settings, they differ a lot in terms of realism and control. In the laboratory setting, we have little to no control. We have a lot of control, but there's little to no realism, which is the opposite of natural experiment. But if we look closely on all sorts of laboratory experiments in cognitive or social neuroscience, we may easily see that, particularly in neuroscience, we're not very much advanced in terms of ecological validity. There's this nice paper by Rita Hari from 2015 about centrality of social interaction, when she sums up the stages of the research. If you looked at the development of the methods in social cognitive neuroscience, in social neuroscience, basically early 90s, very basic perceptual and motor paradigms. After that, people had this idea, maybe let's just show photos of pictures and houses. This is how the, stuff from 97 is. Basic stuff about how do we process static pictures of this or that. And after that, people had ideas like, okay, but in a real social world, people are dynamic. So maybe instead of using pictures, let's use dynamic movies. And this is where we are. For the most of the research in the fMRI field, in the EEG field, we bombard our participants with all sorts of social cues, non-social cues, and look at the brain's patterns of response. How does it relate to the real-life social interaction? Probably a little, I would say. So what does Hari say? She says that the perfect situation would be to be able to capture real-life, unbounded social interaction in the life, in our laboratory setting. But, as you probably will hear a lot for the next few hours and the next day, there are many problems with this approach because there's like a lot of data and little of models available to analyze it. So maybe a reasonable thing to do, for now at least, is to produce paradigms which give us a reasonable amount of control, but also go beyond this single person setup. So maybe use some controlled paradigms to study real-life interactions. And this is where we are in our new project, which is introducing this idea into research on loneliness and prosociality. Just a few more words. Why do we study loneliness in terms of prosociality? The idea that loneliness may decrease prosociality was first put forward by Hannah Arendt, when she wrote that basically loneliness produces self-centered bitterness and destroy human ability to act together toward the common goal. And it resonates nicely with what Cacioppo says about focusing on own self-preservation and decreasing our interest in the well-being of others. And actually, if the loneliness epidemic is there, it may go, if there's actual link between loneliness and decreased prosociality, loneliness may be not just the problem for public health, but the significant obstacle in our way to be able to do anything about any of the crises, public crises associated with society not being able to work together and overcome individual interest, like, overuse of natural resources or any upcoming pandemics. So we decided to go beyond this one-person setup and go towards second-person neuroscience, so-called or dyadic hyperscanning tasks using neuroeconomics approaches and social dilemmas. So we started with meta-analysis. We did meta-analyze over 34 studies with over 48,000 participants, and what we found is that actually there is a negative association between loneliness and prosociality, but it's actually weak. It's less than 0.12 in our whole sample. There's marked heterogeneity of results, and one thing which is missing is non-self-report studies, the studies where people actually measure behavior. So we decided to do some studies in this field, and we started with this nice basic idea of introducing our lonely and non-lonely participants with prisoner's dilemma. Probably all of you know what prisoner's dilemma is, but in case you don't, this is like this very basic paradigm when you have to make a decision. You act toward the other player, and you can either cooperate or decide not to cooperate and deflect. If you both cooperate, you get reasonable payoff, like free zlotys. If you both don't cooperate, in a trial, and you do many more of these trials. If you don't cooperate, you go a bit lower payoff. But what is the most advantageous situation for your payoff? If the other person decides to cooperate and you don't, you deflect. So, in a single trial, you may earn a lot by not cooperating. But the most stable trajectory over multiple trials is to develop a stable cooperation with each other. So we ask our participants to play against the other person that they were introduced in our corridor, and they completed a prisoner's dilemma task. And actually, spoiler alert, they were playing against computer, and the other person was just, our collaborator in our first study. And what we found, once again, surprised us, showing like this marked dissociation. Because whether you look at the cooperation rate in prisoner's dilemma or outcomes, behavioral outcomes with actual help or hard task that was done afterwards, you don't find any differences between lonely and non-lonely individuals. They are there in self-report, but we also supplemented this laboratory study with ESM. 7-day ESM, once again, asking people to report whether they had Pro-Social Opportunities Act and whether they acted upon it. And what we found is that basically lonely individuals had less Pro-Social Opportunity Acts and less pro-social acts, but they also had less social interactions. So the most importantly, they were also less likely to act upon the opportunity. If there was opportunity, they were slightly but significantly less likely to act upon it, to do something for the other person. So once again, do we miss something in our laboratory setting? While people are playing repeated tit-for-tat mechanism for several or hundred and several trials? Probably yes. So now, basically, as we discuss it, we were supposed to start our pilot study tomorrow. We have this perfect, nice hyperscanning setup with EEG, prisoner's dilemma, dyadic dilemma, and we have this nice hypothesis that people may act a bit differently if they actually face each other. We are also going to control for communication, direct communication. We're going to manipulate the conditions, whether they see each other, and we're going to compare the results for actual dyads of people playing against each other with either two lonely individuals, two non-lonely individuals, or mixed dyads with one lonely individual to see whether loneliness does impact your social decision-making in this reasonably controlled but also reasonably naturalistic setting. I think I'm done. Thank you. This is like the very brief presentation which was created on the go yesterday. So, thank you. Questions, issues? So I don't know if it's actually about language use, but they show all sorts of negativity biases. If you ask them about any sort of rating, that's one of our hypotheses of why do we see these patterns which emerge mostly in self-report, is that they probably perceive stuff in idiosyncratic ways. There are studies which have shown that, for example, lonely individuals perceive them. They use like multivariate pattern analysis to show that people who are more lonely tend to perceive themselves as less similar to both close companions and other people, so they may perceive stuff in a all sorts of things in idiosyncratic ways, which is one of the theories that we have to explain these results, because basically, yeah, there are marked differences between self-report and all sorts of fMRI, EEG, and behavioral paradigms we use. So it's not like the sole hypothesis, but this is also something that may be there, and it's further explored as we speak, because there's like a lab which basically focuses on this part of the issue with loneliness.
found that depressed patients are more likely to use the word I rather than we, which is totally
Yeah. So the issue with depression is that basically for most of the 20th century, loneliness was not even studied outside of the realm of clinical psychology, because many people believe that being lonely is just part of being depressed. So I think that there's like a semantic link which may also be there for loneliness. And one important disclaimer is that basically we try to capture people who are very lonely, the loneliness, without clinical comorbidity. It's very hard to do, because most lonely people usually have some mental health problems in our, at least in our pools, because basically if we screen them, they usually emerge as, you know, over the cutoffs for the clinical depression episode, etc., etc. So it's hard to fully dissociate one from another. So basically I would argue that, yeah, they may be also relevant for loneliness, such studies. We plan to have them. We plan. We're only starting. We'll see about that later. That's the plan. That's why we have such a populous sample here, to also have like single-sex diets. We also discuss whether we should also look at the sexual orientation and this type of stuff. But if you try to parcel out everything, it will be very hard to, you know, complete any of that. So for now, we just decided that we'll try to balance the diets in terms of sex, age, etc., etc. But basically, there's one caveat. There are way more females who are in our recruitment samples, because women are just more likely to, you know, engage with our leaflets, with all sorts of recruitment protocols. And even if we have male participants, they are not lonely. Finding lonely males who want to come to our laboratory to do anything is like the hardest part of it. It's not. There's no. And lonely, but there's also way more women in our samples. Probably everyone who's done research in social science found this effect, and this is like nice replicable effect that shows a lot. But if you have this male participants, and if you have cut out, like we have like quartile-based recruitment system, so we want to have like only top quartile, only individuals with no clinical comorbidity, which makes it a bit more tricky. So the first time I got the question whether people vote for this or that right-wing party because they are lonely was around 2019. So why the media focus on specific stuff is beyond my comprehension at this point. But I would say that, yeah, this is like something which is a thing that is often discussed in terms of political orientation, lifestyle, etc., etc. And if you go further, one of the not too many books about loneliness which were actually produced by Polish authors is about the incel culture, przegryw, right? So basically it's not much there in Poland, and maybe we have a lot of information about this specific sample. I don't know. It's why media are doing stuff like that. We would probably have to ask other person than this psychologist, neuroscience/neuroscientist. Called Okruszek. Sorry for that. Any other questions before we go any further? Agnieszka wants to move split forwards. I'm okay with that. Thank you.
I will speak loudly. I can do it.
Yes, you can.
Okay. Okay, so thank you. Yeah, I can. Thank you very much. It was indeed very interesting lecture, and thank you for jumping in. So we owe you the present and probably more. So I owe you also a dinner, but it will be delivered later. So thank you very much.
Much appreciated.
And now we can move on to another talk by Francie. So Francie, you might want to switch the computers. And I am also really looking forward to your results, Łukasz. So.
I'm also looking forward.
And if EG wants work, you can switch into F-News.
Yes. Maybe.
The mic is for you.
Oh, I'll also take this. All right, is everyone ready to continue right away? I know this is a lot of input, but you know, that's what's the great thing about workshops. So hang in there until the next break. Yeah, so it is my great pleasure to be here today to support this really awesome workshop and project. And I kind of hope with this talk to tie in what you heard this morning about FNIRS with the concept of hyperscanning and with the whole idea of this project. And I want to introduce to you how you would perform a hyperscanning study with our equipment, give you a brief overview of the hardware that we have, and also show you just very briefly a couple of example studies so you kind of get an impression of what's possible. This does not work. Okay. Then I will go like this. Okay, so yeah, first off, you've already heard this morning, but just to give a brief recap. So when we talk about hyperscanning, we talk about it in the context of neuroimaging. So we are neuroimaging two or more people at the same time to study the brain during interaction. And I'm not going to go too much into, you know, the application examples. Yeah, sure. I did plug it in, but maybe. Oh, 1 second. Is it like turned on?
Yeah,
Sync this too?
Oh, maybe not.
Yeah. Okay. Okay, very good. Okay, I'm not going to go too much into the application examples because I'm going to show a couple. But rather, what I want to do is I want to point out here that, as we have heard, as it's also the plan for this project, is hyperscanning is just one level of the research that we can do when we look at relational neuroscience, is what the authors of this very recent paper called it. So researching how, you know, humans interact and how their bodies and brains interact when they stand in relation with each other, in exchange with each other. So hyperscanning is exactly where kind of this you know, neural layer comes in. So this is what I want to focus on today. And yeah, we already heard a couple of really great, you know, examples for why it is interesting to employ hyperscanning, but maybe I can bring in a little bit of a different perspective, kind of, I think, also in line with what the project is trying to do. So if we, you know, look at this setup and, you know, we imagine hyperscanning in a context of neurodevelopment and neurodevelopmental disorders, wouldn't it be great if we just had a neural marker? We put a system like this on a child, we have them interact for 5 minutes with their caregivers, and we're able to tell by just measuring the brain activity if there is some sort of atypical neural development happening in the child, be it, you know, ADHD or autism, anything along those lines, and then based on this assessment, we can very early on implement some sort of help for this child. I think this would be a really, important thing, because the earlier we help, the earlier we're able to identify these sort of atypical neural developments, the better we can help and the easier it will be for the child to have, like a good trajectory, how they can develop. So again, this is just as a little bit of, an additional motivation, something that I think would be a really cool application of hyperscanning in the future. Of course, there's a lot of basic research that needs to be done until we are at this point. But I think, this kind of illustrates very well the value that hyperscanning could have at some point in a very practical sort of view. And if you don't believe me, believe this paper. So hyperscanning is very, very popular. You can see over the last 20 years a very steep increase in hyperscanning papers. And not only that, but also when we split after by modality, you can actually see that FNIRS hyperscanning is by far the most popular technology, has been and is still. And there are good reasons for that. And these reasons I want to explain to you a little bit more. I think Dalila already gave a pretty good explanation earlier, but let me quickly recap. So FNIRS and hyperscanning is so well established because it is just very comfortable and easy to apply, and thus suitable for all age groups. So you can measure from infancy over childhood to even clinical groups and older adults. So you're pretty much not limited, and you don't have to exclude any participant population. You can measure participants with implants, for example, cochlear implants, people that you couldn't put, for example, in an fMRI. So this is really, really crucial. You can really assess everyone with the technology. And then, of course, the question also came up earlier. We have a lot of systems now on the market that are wireless and mobile. Also, FNIRS, as we've heard, is quite robust to motion, which allows us to bring our experiments into a naturalistic environment and increase the ecological validity, as you've just heard in the great talk that we had before. It's really, really important to actually get to the base of what makes a human-to-human interaction, not through a computer, not in a standardized way, but really, what is it that makes face-to-face contact with a person so special, and what happens during that time. And that is really, really crucial, because traditional setups might not allow us to get to the bottom of that. Also, FNIRS is, as we heard, quite compatible with other technologies. So you can integrate a lot of, what we saw with the relational, with the levels of the relational neuroscience, bio-behavioral synchrony. FNIRS fits in very well because it integrates very nicely with several other modalities. Now, very quick overview on how the hardware and software works and the solutions that we offer for it. So this is kind of like a complete overview of the portfolio, things that we've released over the years. I'm not going to go through everyone, but this is essentially to show that we're trying to add onto our systems to allow you to do more research in the fields that are requested. So we really try to listen and implement these add-ons to our systems as we go. And the ones that I want to go over for hyperscanning are specifically our hyperscanning app, which we will also see this afternoon in the hands-on session. And then in terms of the systems that you can actually use, we have 3 that we can employ. Kind of the older system that you can see here in the back, the NIR Scout, is our, one of our original systems. It's stationary and wired. So I'm not going to go into too much detail on that, but I want to focus on the NIR Sport 2 in combination with the Wings, which is our physiological monitor. So that's kind of the setup that you can see over there. The white boxes there are the main amplifiers, and you have the cap connected, and that's pretty much the whole system. That connects wirelessly to your computer, and important for hyperscanning, you can use it also on 2 participants. And how would you do that? So there are essentially two options. So you can split up a single device and use it on two participants at the same time, or you can use multiple devices next to each other. And this we have currently tested with up to 10 devices at the same time. And this was only the devices that we had available, so more might be possible depending on the processing capacity of your computer. So that's more the limiting factor there. So you can see you can get really, you can start dreaming about what might be possible in terms of, recording in a classroom, for example, recording from me while I'm presenting to you, recording from you while you listen to me, things like that. Bigger, larger corporations, cooperative setups can be really possible here. And to kind of show you what it would look like, so here you can see some photos from COVID times. But yeah, so this is what a single device setup would look like. So you can see the participants are wired to the same device and they each are wearing their own cap. Everything is recorded together. And here are a couple of pictures of what the multi-device setup would look like. So everything is wired together via an access point. So it's kind of like a router creating its own Wi-Fi network that's connected to your computer. And then you can be completely wireless. And actually in the top right, you can see some of my team colleagues playing laser tag while wearing the system. So this was more of kind of like a fun challenge that we set for ourselves. But again, just kind of illustrates really nicely, what might be possible. So yeah, important feature, very important. And also I've worked with a couple of systems for hyperscanning, and I really, really like our hyperscanning app. It's really easy to use, very surprisingly easy. So it's essentially an app outside of our recording software that we use to control several instances of our recording software. And it controls everything. You can start and stop signal acquisition, and everything runs via this app. It also takes in the triggers from your experimental paradigm, and also, again, can be used for single and multi-device setups, and we'll actually show both of them this afternoon, so you can see how both of these setups would work. And yeah, so an important consideration in hyperscanning, not just in FNIRS, but any hyperscanning setup is to consider that you need shared triggers or event markers in your data. This, the Hyperscan app, you can start a recording in the two Aurora instances, so Aurora is the recording software, at the same time, but you still need to send triggers to both devices to later on be able to align the two data sets with each other. Now, I don't know how familiar you all are with triggers, so quick detour, what is a trigger? So a trigger is essentially a little electrical pulse that you send to, it used to be an electrical pulse, nowadays you can do it via software, but it's a mark that you put in both of your data sets at exactly the same time to mark a section of interest. So you can later on analyze your data with respect to this section of interest. So this an important disclaimer. So we offer this hyperscanning app, which is really, really awesome and easy to use, but you should still send your triggers that you don't get around. But it's really easy with the software tools nowadays. Again, we're gonna see that later. And here's just a little summary of the possibilities that you have. As I said, triggering is always important, but you're really flexible. You can get, for example, one system, so you could use the setup that we have over there with the two boxes. So this could be either a single person whole head setup, like the cap that I've set up there, or you could use them side by side on two participants. And again, you can stack those systems and add up to 10 more of those. And you can also add additional measures, for example, accelerometer or the NEREX wings, which we're going to see for this brain-body behavioral synchrony measures. It's a really nice add-on. Okay, and now for the application examples. So I'm not going to go into too much detail for the single studies, but as Dalila said, we're going to provide those slides to you after the workshop, so you can do some reading yourself if there's anything that piques your interest. So According to this study from 2018, these are the most common task types that you will encounter in hyperscanning setups. So we have imitation tasks, very simple, where one participant tries to imitate the movements of the other, or also synchronizing tasks, so where participants try to synchronize their movements. Those are very, very common. Very simple eye contact or gaze tasks, which is actually also what we're going to attempt this afternoon. The experiment that we prepared. As we heard, something that's super common is also economic games or exchanges, as our previous speaker used. Also cooperation and competition tasks, and then again, very important for FNIRS, interactions in a natural scenario and even interactions during a conversation. And this is really where FNIRS shines, because it's not as strongly influenced by artifacts relating to muscle movements. So you can also, look at the brain activity while the participants are speaking and talking to each other. And I have brought with me as the first example that I want to show you, very old study. So this is pretty much the first one that used our, and you can see it over there, stationary system for hyperscanning. As the authors wrote here, this work represents the first use of a single FNIRS instrument for simultaneous measurements of brain activity in two people from 2011, I believe, or 2012. Where the participants played an economic game side by side, just sitting very closely next to the same computer. But already there, the authors found that the brain activity essentially in the right superior frontal cortices of the participants sort of increased and aligned during cooperation, and that also the extent to which this synchronization of brain activity happened also was associated with better cooperation performance. So this is, I think, kind of like a sneak peek of what we're going to talk about a little bit more tomorrow during the analysis part, something that we can look at when we look at hyperscanning data. So this is just, fun kind of to see where it all started. And we have now gotten much further. So I have a couple other examples for you for FNIRS hyperscanning. Here, for example, during cooperative tasks, and you can really see, so as the systems get smaller, we're able to make the interactions more and more naturalistic. Here, for example, during creative problem solving in teams, with a really, really interactive tasks, participants moving, building things together, interacting, talking. Or here, this one kind of fits in well with the topic of this workshop. Researching mother-child dyads, also freely interacting and playing with each other. And in this study, the authors looked at the effects of the interaction quality on neural synchrony. So they coded also the interaction quality in a behavioral way and then related that to the neural measures. And let's see if this next one works. So here, this is actually also from this exact lab. I want to show you just a quick example of what such a hyperscanning setup could look like. And this is actually, the system they used here is the NearSport 1, so the predecessor to this one, but also a mobile system. So you can see, well, the baby there wasn't a fan of it, but generally it's really fast and really gentle, and this is kind of how a setup with one device could look like, and you can really see like the interaction is quite naturalistic. So just to drive home that point of how well FNIRS is suited for this purpose. And to bring it even further, this study I recently read, I found very, very cool. I'm also super interested in music research, so this was a really nice example for hyperscanning also. A group in Brazil they researched FNIRS responses in professional violinists while they were playing duets. So they had the FNIRS on the head, the participants were playing, and they looked at the brain responses in the leader versus the follower violin. So this is a really, really nice study. A little bit limited because they only had the probes on one side of the head because they didn't want to impact the participants playing. But generally, again, kind of really nicely shows the possibilities that you have. And lastly, because this also ties in with our session this afternoon, and I think also nicely with sort of the topic of this whole workshop, is the approach of integrating brain and body synchrony. So I think this is, this is kind of what you're attempting to in your project. There are more and more researchers who assess FNIRS and physiological signals together and then try to relate those two with each other, and they are trying to see how much does the brain activity or the brain couple with each other and the body the bodies couple with each other, and how does the one influence the other, and how much variance is explained from the one to the other and the other way around. So, and this exact study setup we're going to try to replicate this afternoon. So yeah, and just to close this out, there are a couple of features that I think make our system really nicely suited for hyperscanning studies. And first of all is really the unmatched data quality through hair that you can achieve. And I mean, in every study, it should be our goal to record good data from all participants, and especially in difficult to recruit subject groups, such as, for example, infants and clinical groups and all of that. We can't afford to lose participants due to their hair color. Plus, we also don't want to measure only from one group, and we want to be able to image the whole brain. So this is really, really essential, and it's not an easy thing to do for an FNIRS system. So this is something that we really, kind of subscribe to. We want to be able to measure from everyone. Also, I hope this talk kind of made this clear, how modular and scalable our system is, and that even if you have the system for hyperscanning, you can also use it for other applications, which I think is really nice. Again, also we have cap sizes for all age groups available. So if you want to, start with adult to adult hyperscanning, for example, and then move on to parent-child hyperscanning, because that's usually the trickier thing to do, that's something you can absolutely do. Also, again, kind of ties in with the first point, the montages. So how you place your sources and detectors on the head is completely flexible. So you can measure from every part of the head, and you can also switch between studies. So in one study, you might want to target the prefrontal cortex, in the next one, the parietal, occipital, etc. Also, and Dalila kind of mentioned this at the beginning, is that we place a big emphasis on support, and I have a couple more resources to show you at the end. So we really put a big emphasis on enabling you to do the best research you can and to support you in that. So we have a lot of webinars and tools to help you get to the point to be able to analyze your data and publish. And again, with this workshop, we kind of also hope to contribute to that. Already mentioned the possibility to integrate physiological and other measures, and also our strong track record in peer-reviewed FNIRS, but also FNIRS hyperscanning research, I think is a very strong point that shows how well suited our systems are for this kind of application. For the further resources, don't fret, there are a lot of reviews out there on FNIRS hyperscanning and other hyperscanning. It is a very, very popular field. So if you're only diving into it, these are a couple of the resources that I can really recommend. And again, I think we are going to dive into this a little bit more tomorrow, but if you're interested in the analysis part, which, is kind of, as we heard, a little bit of the, we know how to measure all the signals, how to analyze it is more of the question. But if you're interested, I can really, really recommend this review, which also goes over, like how the different approaches work, which ones are the most commonly used. So that's a really great starting point if you're interested in that. And I do want to point out also our 2 webinars that we held, I believe, last year with Pascal Vertica from the UK. He's a really, really good FNIRS hyperscanning researcher, and he did 2 webinars for us on the underlying theory of FNIRS hyperscanning and the analysis. So these are really, really great starting points for you if you're just starting out. I know there are a lot of resources, but it's a really exciting field, quickly growing field, I think, as the graphs in the beginning showed. And we're really excited to contribute to that field and to help you do the best research you can. And that is it from my side.
Mm-hmm. And isn't it difficult, I guess it is, to divide each of those movements and to find the exact part of brain activity that is, let's say, the goal of the study?
Absolutely. I think that's kind of what was hinted at in the previous talk. It's kind of trying to find that balance between making your experiment as naturalistic as possible while still being able to draw something from your data. So I know there's this whole discussion about how to effectively design a hyperscan experiment. It's also very difficult, for example, to find control conditions for hyperscanning. That are effective. So what most people do, I think, is they have kind of like a semi-controlled setup. So for example, I think you were referring to the task in the teams, right, where the participants had to build something. So I think I would have to, possible that I don't have all the details in my mind anymore, but I think what they did is it was semi-controlled in the sense that they told the participants, okay, now you start with the interaction, and they gave sort of certain time segments which they then marked in the data, and then they had behavioral coding in the middle, which is, I think, the approach that is most commonly used. Is you videotape the interaction, and then you have certain markers in, or let's say you qualify the video based on certain criteria that you set before, and you try to kind of course, let the participants roam freely, but maybe you set some restraints, like some, to just make it somewhat comparable between groups. But it is very challenging, for sure. Yeah. Any other questions? Okay. Oh, yeah.
In that same direction, what's the most common methodological approach at the end to compare, right? Because Dalila was saying all about the block, the resting state,
Mm-hmm.
but what is the most common one used in these studies where it's more ecological environment?
Right. I would say for sure functional connectivity is something that a lot of people use. Of course, also, motion artifact correction, the correct approaches are really, really important, because the more, movement we have, we can try to minimize the motion artifacts as much as we can, but they will still happen. So it's really, really important to consider that from an analysis point of view. And then there are these different approaches to measure actually this connectivity, right? Like coherence, correlation, all of those different ones, which are really nicely outlined. So the most common one is coherence. But for example, this other study that I showed with the violinists, there they actually had kind of like more of like a block design where they had, blocks where the participants played as like duets and then solo, and then compared the activity within those blocks with each other. So there's a lot of variability, but I want to say that the most common is for sure, like this functional connectivity and this freely interacting thing, and then looking at certain time segments in your data. Okay. I think that's it. Perfect. Should I turn it on? Is this how I turn it on? Okay.
Thank you very much. It was really great. So again, thank you. We will still meet a lot today, yes, but it's a small incentive. Stay so with us.
Thank you.
And now it will be a little bit tricky part because we have to switch computers. And make sure, so Jean, we are now switching computers, and shortly you should be visible. Oh, perfect. One more thing, I will just make sure that I am plugged in. Yes. And Jean, can we make sure that we can hear you and that you can hear us? All is good? Okay, perfect. Okay, so I will introduce you, and it's a great pleasure and honor for me to introduce Jean, who is not only a researcher. I think you are now an institution in this, research in the field of bio-behavioral synchrony. I think that Jean is co-author or the first author in the majority of papers in this field, so I am really glad that she joined our group. And today she's going to give us a talk on interpersonal neuronal synchrony in parent-child dyads. So yeah, Jean, the screen actually, not the floor, but the screen is yours.
Thank you very much, and thank you very much for the invitation. I don't know if you recognize me, but yeah, so first of all, my name is Chin Yuan. I'm a postdoc at the University of Heidelberg and Italian Institute of Technology. And the person in one of the videos was actually me at the beginning of my PhD when I was young and naive. And now I'm less naive, I hope so. But I'm very happy to actually bring you a selection of the studies that we did in Leipzig as well as at the University of Vienna where I did my PhD.
I will just make sure, because I think that I can hear you well, but I'm not sure about other people. Can you hear? Not really. So I will just ask IT team who can hear us to maybe do something with audio. They waved to me, so probably they can do something. Because I think I can hear you from my computer, and we should be able to hear you from loudspeakers. IT staff, can you hear me? So they are sitting in, okay, they are waving, so hopefully, I think they are coming even. Because,
say something.
Can you say something, maybe? Maybe they improve it?
Is it working now?
No, still from a computer, so maybe they will come. Maybe we can, it's a very sophisticated room we are in. A lot of technology.
Maybe too sophisticated.
Maybe. Oh no, I think we did something. And now it's working. So yeah, can you
Okay.
hear Jean? Yeah. Yeah, I think it's better. Okay, you can continue.
Okay, because I can't like hear anybody else in the room, but I...
Yeah, it's a big audience. Then I can show it to you, but yeah. Okay.
Okay. All right. Then I'm just going to continue into the presentation of our studies. So what drove the studies that we did from the very beginning is that social interactions are a main driver of child social, cognitive, and emotional development. As we all know, there is a majority of theories behind this phrasing, all the empirical evidence that has been there from the very beginning, saying even in the 60s, 70s, 80s, like when like Dan Stern and Jaffe, for example, have done this kind of work and have looked into the role of the child and the caregiver in interactions and what it does for child development. So we try to do or take a similar approach in this by first looking at the child. And here we can kind of identify this very early capacity or this propensity for making predictions from very early on that can be at birth, that can be also at 2 months, depending on the capacity that we're looking into. And what's really interesting is that now more recently, many of these behaviors have been explained by the predictive processing account, or at least put under this predictive processing account. And you can see this, for example, in this baby on the right. This is a study by Reddy et al. From 2013, where the baby is picked up by their caregiver. And they're lying on this mat with loads of different sensors. And what the authors realized in this paper, or in this project, is that the baby, starting from 2 months of age, start to prepare their body to engage in this like picking up situation. So they make a prediction of what to do next when they're interacting with their caregiver. The goals of this prediction making are manifold, and they can be to minimize prediction error, right, to make the interaction as smooth as possible. But then it's not just about making precise predictions per se, but also making useful predictions. So when is it good to make a prediction? So the predictions need to be weighted. So this is from the child's perspective. But then during interactions, we also have the other side, which is the caregiver. And the caregiver, from what we know and a lot of different research, is their propensity to enhance predictability through communicative rhythms and contingency, right? So the caregiver most often just doesn't sit there and engage with an infant like with another adult, but they engage in processes such as joint attention, affectionate touch. They use music, such as infant-directed singing, or rhythmic movements, such as bouncing or rocking. And when these two come together, we can see something that doesn't occur all the time, but in some instances, and that is social coordination. So this term, I mean, it's not just one term, right? Like there are many different terms, and we've heard about it yesterday as well. It's also called interpersonal synchronization, and this is defined as the dynamic process by which hormonal, physiological, and behavioral cues are exchanged and adjusted. So this is one of the many definitions. And synchrony per se is not just concurrent synchronization, where, for example, looking at each other, mirroring each other's gestures, postural sway, but what we also mean by synchrony is its sequential aspect, meaning turn-taking, reciprocal actions, and imitation. So more recently, this has been looked at not only from a behavioral point of view, but from a multimodal point of view, meaning physiology and endocrinology at the lower levels. And then, I mean, we can look at it as a hierarchical model. And then if we go higher from a neural level, as well as from a higher cognitive level as well. So these are just some examples that I kind of substituted from the original paper. They had different examples, but this is very much the same. And this is something that we started looking into in our earlier studies and are still looking into today. So I don't think I need to define interpersonal neural synchronization anymore, because you've heard about this many times now, and about hyperscanning. But I think what I want to emphasize still is that we're looking at interpersonal neural synchronization as the alignment of brains that is marking, however, like mutual alignment in behavior and physiology, right? Like we're not talking about telepathy or any magic. We're talking about marking processes that are actually happening in the environment, or information that is transmitted through the environment. And what has been suggested in many different frameworks from different people is that enhanced mutual prediction of actions, of each other's actions, and shared attention towards each other or some common goal is enhanced, or is related to higher interpersonal neural synchronization. So the link is that social interaction stimuli are optimally processed when we synchronize our brain activities. So coming from this question, we wanted to ask, how does this look in early parent-child interactions? Like, can we find some evidence in that domain? And there have been like some people leading this work, like Vicky Leong and Sam Ross, for example, or Vanessa Reindl and Kerstin Konrad. But we wanted to take our own spin at this by looking at it in a very naturalistic interaction. So in our very, very first study, which we did at the Max Planck Institute in Leipzig, we asked this very simple question, or relatively simple question, it's not that simple, right? In a mother-child diet situation where they were solving a problem together. So we had 42 mother-child dads come in. The children were 5 to 6 years of age, and they engaged in a cooperation condition, individual condition, and a resting phase. So we tried to mirror what we knew from the adult literature. And we measured neural synchronization using FNIS and had video recordings to then rate reciprocity and child agency. And maybe to kind of apply the knowledge that we just gathered from the NIRX team, this was done using the Near Scout device, so the stationary device. So I'm also not gonna go into the details of what FNIS does. You all know by now what it does. But what I wanted to mention is the areas that we were looking into, or that we're looking into in all of our studies. And these concern the prefrontal cortex, the PFC, the temporoparietal junction, and the inferior frontal gyrus. And all of these brain areas have been associated with interpersonal neural synchronization in adult studies, but also in adult child studies, and are also associated with social processes that are very important to interacting as smoothly as possible. And in terms of how we calculated INS, or interpersonal neural synchronization, is that we use wavelet transform coherence. So that was previously mentioned as well. It's a period matching measure, meaning that we can measure signals that are in phase, anti-phase, and phase lagged. So it's a very liberal measure in that sense. There are much stricter measures where you have to be, for example, in phase, meaning that both signals have to rise at the same time and fall at the same time, but this is not the case for coherence. Okay, so methods now done, we can move into the results section. And what we found in this very first study is, yes, indeed, we can find higher neural synchronization in the cooperation condition in comparison to our two control conditions. And what we then did is try to look closer into the cooperation condition. So how did the parent and the child cooperate there? Like, did they really take turns talking to one another, showing positive affect, being considerate of one another, or were they just like doing everything kind of by themselves and then putting the puzzle together at the end? So we have this behavioral reciprocity scale, the higher, the more reciprocal, the lower, the less reciprocal on the x-axis, and then on the y-axis you have WTC, so neural synchronization. And what we found here is that the more the diet behaved reciprocally, the higher neural synchronization was in those diets as well. So the individual variants actually covaried across these two variables very nicely. Next, we also wanted to look into the individual behaviors of mom and child, like did those behaviors in any way drive neural synchronization as well? And what we found when we looked at child agency, so how much freedom did the child have to behave as they wanted to? We also found a positive correlation between neural synchronization and child agency. And what you can already see here is that the slopes of the correlation are not that big, right? And this is, I think, a very consistent story across all the different data sets that I've looked into thus far, is that the effect sizes are not that big. But still, we explain some of the variance that we can see on the behavioral level as well. So what we can see from this very first study, in summary, is that we can find some evidence on the relation between interpersonal neural synchronization and interaction quality markers. So next, we wanted to dive a bit deeper into those interaction qualities. Like, what does it mean exactly if they're behaviorally reciprocal, or if the child shows more agency, right? And in this very early study, we didn't have the capacity to do microcoding, meaning manual labeling of frame-to-frame behavior, and this is what we then did in the next study. Well, we then also moved into mother-infant interaction, because this is where, at least for us, the majority of the microcoding work from, for example, Beatrice Beebe was also done as well, and so we used her paradigm as an inspiration for this study. So what we did here is ask this question: are attachment theory-based predictive behavioral patterns, so such as touch and proximity, related to neural and physiological synchrony? And here we invited 69 mother-infant diets to the lab. The infants were 4 to 6 months of age, and this was already, this was done in Vienna. And here we used the Near Scout, and this is from the study, or the video that you actually just saw, so this is the study behind it. We had 3 different conditions. So we had 2 control conditions in a way, which were the distal watching and the proximal watching condition. So in the distal watching condition, mom and infant were sat next to each other, and they watched a calm aquarium video for 90 seconds. Then in the proximal watching condition, the baby was seated on the mom's lap instead to increase proximity. And then in the free play condition, which always followed the 2 watching conditions, mom and baby were instructed to play as freely as they normally would at home. And here is when we actually coded the behavior of mom and baby frame by frame. Again, we measured neural synchronization using FNIS and wavelet transform coherence. And in addition, we also measured physiological synchronization of ECGs in both mom and infant, and used cross-recurrence quantification analysis to calculate interpersonal physiological synchrony. And then we measured the behavior from the video recordings. And in this particular case, we dove a bit deeper into maternal touch and mother and infant affect. So in terms of the results, what we found is what we actually hypothesized as well, but a bit more complex, as it always is. We found the highest levels of neural synchronization in the free play condition, but only in the prefrontal cortical areas and not in the IFG, which is normally found in adult participants. And then the other differentiation, what we found as well, is that the proximity actually already enhanced interpersonal neural synchronization between mom and infant, as you can also see by the yellow box plot here, which was also increased in lateral and medial prefrontal cortex in terms of neural synchronization. Similarly, but also slightly different to neural synchronization, we found that moms and infants showed higher physiological synchronization but only in the free play condition, and we didn't find any increases in terms of proximity manipulation for physiological synchronization. So next, we wanted to dive a bit deeper into the behavior, right? Like, as I told you, we wanted to do these micro codings to see, okay, what are the behaviors really associated with neural and physiological synchronization? So what we did next is look at these free play behaviors. We first looked at affectionate touch, and what really nicely mirrors the proximity results from earlier is that also affectionate touch differences in the diets in the free play condition were related to differences in neural synchronization. So the more affectionate touch the mom showed, the higher the synchronization in the brain activities of mom and baby. we didn't find this correlation for physiological synchronization, but we found instead is that which seemed to be indicating this kind of co-regulatory effect, is that the more negative affect the infant showed, the higher the physiological synchronization. So while neural synchronization seemed to mark this kind of more maybe communicative instances of the interaction, we found this more co-regulatory effects in physiological synchronization. So there might be some differential pathways for neural and physiological synchrony, but this is just beginning, and we definitely need to dig deeper into this differentiation or this correlation between neural, physiological, and behavioral levels. So for now, we looked at the neural and behavioral levels and the correlation between each other. And for the following studies, what I'm going to do is move one step higher in terms of this model, where we're going to look at neural synchronization and higher level cognitive systems and how they are related. So once we knew about this relation between behavior and neural synchronization, we wanted to look at what the diet would bring to the interaction, right? Like they have a history, they have experiences that they've gathered, and that would inform this relationship that they have with the person they're interacting with. Like, as you can see on this model, Simone Shamaitsuri, is that the more you interact with one another, the more expectations you have about like how to interact with the other person and how this interaction might pan out even in the long term. So there is this assumption that potentially functional similarities of interactants' brains might facilitate interpersonal synchronization. There is growing evidence on that, but I think there are still lots to do. This can come from a genetic point of view, of course, right? Like if you think about like the behavioral closeness, the biological closeness of parents and children or grandparents and children. But then there might also be some functional factors, and there a lot of other people have looked into, for example, the role of friendship in similar brain patterns, as well as the role of being relatives or family members when you react more closely in terms of physiology or neural activation patterns as well. And if we think about it from a more cognitive point of view or even attachment theory-based point of view, one might assume that overlapping mental models might also result in similar intra- and interpersonal neural responses as well. So we were especially interested in the attachment theory point of view on this, so this is what we did for the next study. So first, from attachment theory points of view, there is more than one caregiver, right? Like fathers are often neglected, as we heard yesterday as well. And I think because one of our co-authors, Pascal, is also a father, like a very engaged father, he wanted to make sure that we take them into consideration as well. So what we did is try and first replicate the same results as we found with the moms by inviting the dads and doing the same task. So we invited 66 father-child dads, again 5 to 6 years old. The study was conducted in Leipzig again, and we just reran the whole study as we did before. Cooperation, individual problem solving, as well as resting faces. We again measured neural synchronization and had video recordings of reciprocity and child agency. But in addition, we also wanted to first use this opportunity to dive into the relationship quality of the fathers and the children. And we asked the fathers how much they were involved in child rearing. So were they really engaged and knew about their child's schedule, how much time did they spend with their child, did they think they should support their child, all these kinds of different caregiving beliefs. We were asking them about that. So in terms of the results, we can see that we can actually replicate, or we replicated the mom's results in that way. So they showed, the father-child diets showed higher interpersonal neural synchronization in the cooperation condition in comparison to the individual condition and the rest condition. But what was very puzzling to us is that none of the behavioral results actually replicated in the DADS, and they don't replicate to this day. So whatever I do, we just don't find anything, which is, I think, very interesting. And if you have any counters, like pointers, let me know. But what we found instead is that father involvement was related to neural synchronization. So on the scale of being a supportive and warm father to the child, we found that the higher the belief of the fathers on that scale, the higher the neural synchronization with their child as well, which is a first preliminary evidence to the relation between INS and father-child relationship quality. But this, of course, is just a very coarse measure. There are better measures in terms of relationship quality. So we then didn't, and to amp up this whole situation, is to use proper attachment measures, such as the adult attachment interview and the story stem procedure for the children, which are the two gold standards for attachment measurements at these different age points. So now we use those 66 father-child dyads and additionally invited 52 mother-child dyads to do this kind of cooperation task again, measured all the things that we wanted to measure, and in addition, we also did a scaffolding task for the parent and the child, where the parent was instructed to just help the child do this task, but not interfere if in any way possible. So in this way, the child was kind of leading how much they wanted the parent to be involved. So here, what we first found when we looked at parental attachment representations is something that we actually didn't expect. So we actually found that insecure attachment representations in the mom were related to higher neural synchronization in the mother-child dyads. But then when you think about it from a more behavioral point of view or attachment theory research point of view, it might actually make sense that the insecurely attached mothers might try to engage the child more because they're hyperactivating in their behavior to kind of compensate for their cognitive differences or representational differences with the securely attached mothers. Exactly, and this is, still this has to be taken with a grain of salt because it's only in one area in the lateral DLPFC and only in the moms, we didn't replicate this in the dads. And then when we look at the child attachment representations, we actually didn't find any correlation with it in the cooperation task. But what we found instead is a positive correlation between child attachment security and mother-child INS in the scaffolding task. So when the child was leading the interaction, that's when we found this that attachment security was actually related to higher neural synchronization. And this is what you can see here with this red slope. The slope of the father-child dyad seems to be a bit negative, but this wasn't significant. And supporting this this role of child attachment representations in the scaffolding task, we also found that the quality of the assistance in the behavior of the moms was positively correlated to neural synchronization as well. So it really seems to be the case that this attachment security paired together with the quality of the assistance or the quality of the scaffolding of the mom were related to higher neural synchronization. So overall, we had a look across all the different modalities that seem to be related in one way or another. And I just want to quickly summarize our results for today. So first, we have synchrony and attachment, neural synchrony on attachment on the higher level. And then we have correlations between synchrony on the brain and on the behavioral level. And we find first indications for the role of relationship quality in terms of how much or how high the interpersonal neural synchronization is in parent-child dyads. But then we find much more robust evidence for synchronization and correlation between synchronization in brain and behavior in face-to-face interactions, in reciprocal interactions, in proximal interactions, especially including touch. But then we have this weird result at the end that I just mentioned, right? What about the insecurely insecure attachment representations of the moms. So when we thought about this, we actually went back to some of the earlier work by Beatrice Beebe and Jaffe and Isabella and Belsky, where they talk about this optimum mid-range model, where we have like a lot of it that is in the middle, and I think that's what we're mostly assessing in our parent-child diets that we're measuring and that we invited to the lab. And sometimes we maybe go up here or go down here, and I think this is when we're looking at maybe insecure attachment representations, right? Like where the behavior then tries to kind of compensate what we're seeing to get back to the optimum mid-range that we're mostly looking at in terms of our diets. But this is just, I think, an idea that we're still trying to explore further in our upcoming studies. So for now, I think I really liked what I also heard yesterday in terms of that we need to assess synchrony in much more diverse samples, and I think this consortium is really like the start of something or the continuation of this line of research that is really, really important. I'm very happy to continue working on this project together with Sabina Paun from the University of Heidelberg in terms of the developmental consequences of synchrony. I think that we're still missing a bit as well, like what does it do to the diet, like what does it do to the child? And then I'm still continuing my work together with Giacomo Novembre from the Italian Institute of Technology in terms of how is this coordination visible in early musical interactions. So if you have any interest in that, feel free to contact me as well. So for now, I just want to acknowledge all the helpers and collaborators and co-authors and funders that have made this work possible. Like it's a lot of people, and as you already know, or as you might know, get to know, hyperscanning is tough work, especially with children and infants. So lots of people always need it to do this kind of work. Okay, so thank you very much for your attention. I'm looking forward to questions.
Thank you so much. It was really inspirational and very interesting talk. So do we have questions? So maybe I will start with my questions. So what are your intuitions relating neuroatypical samples? So it is reference to our yesterday conversation about how to involve neurotypical samples and what we can expect and what does it mean. Do we have a good synchrony or optimal synchrony, or what does it mean? So do you have any thoughts on that?
Yeah, I think that's a really interesting question. I mean, we've started, or at least some other groups started with atypical, or gosh, how do I say this? Maybe subclinical states in the caregiver, and this is where we find some first interesting evidence, right? Like, for example, anxious moms would over synchronize with their children, and I think that's something that we might see as well in the diets that we look at. But then when we look at the child, for example, I think it's going to be a bit more complicated. Because there are some assumptions about preterm born children, some assumptions about autistic children, and I think it really depends on the diet and the communication modality. Mm-hmm. So for example, just because you're born preterm and you might have been separated from your parents very early on, they have a lot of coping mechanisms, I think, that can counteract lower synchronization that one might expect in the beginning. So yeah, I think I don't have direct hypothesis towards it, but I think I'm just thinking about all the other variables that need to be collected to make it more meaningful when looking at these different samples.
Mm-hmm. Thank you so much. We have a question. So we have a mic and another mic. You have to switch it on. Yeah, we've got a question from the audience. So,
so hi, Julia here. So when I was listening to it, I was thinking that it seems like synchronization, the term that you use, is not always about providing like good outcomes for the relationship and interaction. Like when you showed us that it can be too much of a synchronization. And I was thinking that maybe we should differentiate between like synchronization, similarity, and maybe coordination and synchrony. Because I was thinking that synchrony in my mind is about attunement, is about co-regulation, is about keeping the quality of the interaction on the optimal level, like for both sides, for both partners of the interaction. So it supports and maintain like some kind of a homeostasis. And what you just showed, I was thinking, okay, so there's too much coordination, maybe of movements, of gestures, or utterances, of something like that, and maybe that's different from synchrony. Like coordination, maybe it's different from synchrony. It depends how you define it, of course. But that was my thoughts, because it's not typical to think that synchrony that it may be too much of a synchrony between partners. Like synchrony sounds like something that we achieve, that we aim for to be like the highest possible, like on intuitive level. We think of it that way. So what do you think about like the definitions, the difference between coordination and synchrony, attunement?
Did you hear it well, or should I rephrase?
I heard 80%. I'm trying to answer, and you correct me.
Okay, sorry, because we are like, the computer is wired to so many cables. I am afraid of moving it around. So basically the question, as I understood clearly, I will try to rephrase to make sure that we are on the same page. So what's the difference between coordination and synchronization? And whether better synchrony leads to better outcome or not necessarily, more or less. But we can start with the distinction between coordination and synchronization. Yeah? Okay. And then you can use that 80% you had.
Yeah, okay. So I think in terms of definitions, you're like being mixed up and like being a bit fluffy. I think you're totally correct. Like, this is, I think, an issue of different fields coming together. So from what I noticed is like generally, I think we can refer to coordination as the big thing, and there are different types of coordination, and synchrony is might be one type, right? And then I think we can look at, like, if we want to talk about synchrony from a conceptual level that you might have mentioned, I think, with homeostasis and co-regulation. I hope I understood that correctly.
Yeah.
But then we can also refer, yes?
Yes.
Okay. And then we can refer to synchrony, I think, from a more computational point of view. And I think these two are actually very different, right? Because you can compute high synchrony, but that can be conceptually good or bad, like that's a different thing. So what we need to do, and I think we had a chat about this as well in the consortium, but also with different collaborators, is that we need to try and come up with good definitions for different things, and try to think a bit more thoroughly about the methodologies that we're using to compute synchrony. And I think Andrea is going to talk about it tomorrow, if I remember correctly.
Yes.
And there are all these different ways to compute it as well. And then how do we bring it together? I think that's definitely the next step, where the field is discussing quite a lot about it, is the only answer I can give for now. But you're absolutely correct. Like, it's a lot of different things coming together, actually.
Yeah, thank you for your insight. That was what I needed.
Okay, great. Any other questions from the audience? Yeah, we have. I think you can also come here closer, so you will have more social contacts with us. We heard about loneliness, so let's, come.
Perfect. Hi, Chin. I'm Franzi, from New York, so I just did the talk. So I was really intrigued. I mean, this is something we're struggling with in psychological research in general, that the samples we pull are always, highly selective from, people who participate in our studies. And you showed this, this bell curve of attachment styles that we mostly get, people that are in the middle. And I saw that part of the consortium is also to, and I guess this is kind of a shared question, and I wasn't there yesterday, but I'm just curious. Part of the consortium is also planning to assess a more diverse sample.
Yeah, especially in Warsaw and also Milan. So they will have preterm, we'll have preterm and autistic as well.
So I was curious if you also plan maybe in your research to try to get to those other attachment styles more, and if yes, if you have like a strategy for that, because I would imagine it's not super easy. So that's my question.
Yeah, so maybe from my point of view, and then I think Agnieszka, you can talk about your point of view as well. So I think my dream, so this is not a reality yet, but my dream is to have a little van that doesn't look suspicious, but looks very child-friendly and nice, right? And drive around in the different neighborhoods where we know that might be a bit more difficult, and have the mobile FNIRS, like the NIR Scout 2, I think is very suitable for that, to measure those diets then, because they won't come to our labs. I mean, this is not going to happen, right? Whenever we call them, because we do get them from the hospitals as well where we recruit, they don't want to come because they're scared, they don't have time, they have other things on their mind. So I think what we need to do is go to them instead, and try and recruit there. But as mentioned, this is not I think, reality yet. I know of a van here in Heidelberg as well, where somebody is actually trying to do that. And I think that's very commendable, because I think that's the only way of going at it. Either that, going to the families' homes, or going to the kindergartens where we know, okay, it's like a public kindergarten, right? And then you can measure either diets, trieds, and groups, as you just presented as well.
Yeah, right. That makes a lot of sense.
Yeah, definitely it would be a dream to do something like that, but maybe, it will happen at some point.
Yeah, for sure. No, it would be really nice. And I know it's also something that maybe, I mean, you're also already collaborating with hospitals and all of that, but I think trying to start as early as possible to get an understanding of, this is what we're doing. It's not scary. It's not, not for any bigger purpose, but I feel there's that's something that we often encounter as people struggling with kind of the lack of understanding almost because it's very scientific what we do. Not everyone has that previous knowledge, right? Not everyone knows what the data is used for. It's a big thing in Germany, right? Yeah. People are very afraid what happens with the data and things like that. So yeah, trying to start as early as possible I think is very good, and also directly going into the community. So yeah, it's awesome.
And also it would be great to know what to tell parents and how to explain our data, because we won't tell them, you should synchronize your right DLPFC with your kid's DLPFC, because probably it will be good for his outcome, but we're not sure yet. So, but then maybe, touch is also important. So you should touch as much as possible, but not too frequent, because then it's awkward. Yeah. So how to explain our research into something that is impactful and important. So, yeah.
That's an awesome goal. It's a really cool part of the project that I very much look forward to seeing.
Oh, we too. Okay, do we have any other questions from the audience? Yeah, we have one more. Yeah, Karen, you can come here. So we have also a lot of students and researchers here.
Hello. I don't know if there's an answer to this question. As you mentioned, we have to still do research in terms of the developmental consequences of synchrony. But when you mentioned this over-synchronization in samples of mothers that are insecure or anxious, I was wondering maybe what you think the psychological or developmental implications of this over-synchronization could be. Whether this is beneficial or rather not.
That's a very good question. So we try to speculate about this as well, because from an attachment theory point of view, this is where people might think about attachment transmission, right? And then it might not be like, I mean, oh gosh, I don't want to, like, I want to be quite careful about calling things functional and dysfunctional. But yeah, so I think like what I know from my attachment colleagues is that this could be like one way of transferring attachment representations from the parent to the child. So in this case, if it's like insecure attachment representations, one might think that this might be dysfunctional. But we, of course, don't know about it yet because everything that we do has been correlational, cross-sectional, like no long, like, yeah, we don't have any like proper longitudinal studies or proper outcome measurements yet. So we don't really know if it's like functional or dysfunctional in that way.
Okay.
Yeah,
Thank
we just
you.
know
Well,
it results in similar behaviors, like similar levels of reciprocal behavior in terms of insecure and secure attached mothers.
Okay. Thank you.
Okay, thank you very much. Any other questions? No, I think we are done. So thank you very much. It was indeed a very interesting talk. Maybe now I will just show you the audience. So if we disconnect, it won't be a tragedy and you will have an impression that many people listen to you. Can you see? So yeah. Bravo. Okay, so thank you very much, and the present time will take place in Heidelberg because you are not with us here. We are very sorry. Yeah, so this is the end of this official part, and then we will still meet during the workshop. So this is the time for the break and lunch. So thank you very much, and all the speakers and audience and Nirex company and also Warsaw team who made this event possible, and of course European Union who made this grant possible. So thank you very much. Thank you. Bye.
Bye, thank you.
Okay, according to the schedule, we have time until, Eva, can you help me? With the 2:15, yeah, so we have time 2:15? Okay, 2:15, so not so a lot of time. So people who were welcome to the workshops have 45 minutes.